Good morning. This is Wednesday, October 22nd, 7th. We are on to our second part of the muscle lecture. So again, like I said, for the rest of this time, we are going to do muscle physiology during our lecture today, doing the second, and we'll do the third part of the muscle contraction today. And then in lab, we'll move on to our origins, insertions, and actions that control the trunk of the body. So that'll be our game plan. You still have a handful of homework assignments left, 2 unit reviews, the Physio X, and that muscle activity handout. I had a couple good questions during office hours on Monday about that. Remember, when you have a muscle that has 4 actions to it, make sure your activity includes all 4 of those actions. By the same token, if there is a common activity that I've given you that does only 2 of the 4 things that a muscle does, then there might be a better muscle for that particular activity. Some of these activities may have more than one muscle that would do it, but usually there's going to be one that will be the best. So I tried to keep it from being too ambiguous, but it is possible you could have other ones on there as well. And again, remember that one, make sure you're putting some time and thought into it because that one will be graded for correctness. All leading up to a Wednesday lab and lecture exam. Again, that means that the weekend of the 6th and 7th is your last big weekend to study for that. It also means that we have 5 days between the end of the lecture and the next class. So rather than giving you a 5-day holiday, especially with Thanksgiving right around the corner, you will have a couple pre-labs to help you get ready for the nervous system that will be due as well. All righty. Questions on any of that? All right, then let's pick up where we left off. We left off and we were talking about the process of contracting a muscle. And actually, now that I've done this, let's actually go back to the whiteboard. Let's see what we've retained from the last class. So for the muscle contraction, what is the first phase?
A decision.
Okay, that's where it starts. That's the first thing that we're going to do.
Communication at the neuromuscular junction.
Excellent. Phase 1 is, or the first process, maybe phase isn't the good word. Excellent. Communication at the neuromuscular junction. And remember also, anytime we're talking about these processes, it's probably good to think in terms of what its goal is as well. And what is our goal of communication at the neuromuscular junction?
To convert the neural action potential into muscle action potential.
Excellent. As we mentioned, the first step of that is we decide to contract the muscle, and I guess we could be a little bit more specific on that. And what was the last step? What was the last thing we did in the process of communicating at the neuromuscular junction?
You stop, like what decision?
Well, okay, that is how we relax it, but I'm talking about initiation. So when we're talking about initiation, when we're talking about getting the muscle to contract, what was the last step in that communication at the neuromuscular junction? What was the last part of it that we described and identified? Again, this is a perfect opportunity to think back to what our goal is basically to convert a neural action potential into a muscle action potential. Right, excellent. So Sarah's got it. So our last step is that we produced a muscle action potential at the motor end plate. And remember, when we make that big positive wave, is it going to just stay right where it is? When I throw that rock in the pool, does the wave just stay there? No, it's going to spread. And the muscle action potential spreads down the sarcolemma. I make a point of emphasizing this, that our last step of part 1 is producing that muscle action potential at the motor end plate and spreading it down the sarcolemma, because not only is it the last step of the first process, but what else is it?
The beginning of the next process.
Exactly. It's going to be the first step in process 2. And what is process 2?
Coupling.
Say again?
Coupling.
What is it that we want to do second? We wanted to couple something, right? Bring two things together. There we go. Sarah's got it. So excitation, contraction, coupling. And again, we always want to think in terms of the goal. What's the goal here of step 2?
To allow myosin and actin to interact.
Sure, we want to bring the myosin and the actin together, but what's stopping them from being together, right? They're true loves. They always want to be together, but they can't always. So what stops them from being together?
Proteins.
Yeah, those regulatory proteins, absolutely. We need to move the regulatory proteins so the myosin and actin can interact. And if we even want to be more specific, how are we going to move those regulatory proteins?
Introducing calcium.
Exactly. We're going to use calcium to move the regulatory proteins. So myosin and actin can interact. And the first step of that is going to be where we left off with process 1, producing that muscle action potential at the motor end plate. All right, so let's do that. All right, excitation, contraction, coupling is step 2. And as we just said, our goal is to use calcium to move the regulatory proteins. Excellent. Once again, we need to start first with some anatomy. Now, up to this point in time, we have been focused at, oh, no, way too big of a line. Right, everything that happened before happened at that neuromuscular junction. But now we need to expand our anatomy. This, of course, is our sarcolemma. And remember, our sarcolemma is just a fancy name for a plasma membrane. But the reason we give our plasma membrane a special name, as we talked about, is our sarcolemma is specialized to produce and propagate an action potential. Now, propagate's a fancy word. What does that mean again?
Produce?
Well, it produces it, and then what does it do?
Takes it
Remember,
back.
spreads it. Remember, it spreads it. Propagate just means to spread. All right, so if you're propagating a species of corn or something like that, you're spreading it from one area to the next, right? So propagation is spreading. And so that's all it is, just a fancy way of saying spreading. We know one other special thing about the plasma membrane of our muscle cell, skeletal muscle cell. It has these invaginations that travel deep down into the muscle cell. And what do we call those invaginations of the sarcolemma? Yeah, there you go, T-tubules, although be careful. The first time you describe it on an essay question, for instance, you want to describe it as transverse tubules, but then you can always abbreviate it T-tubule after that. Which reminds me of another good point. When you have a big essay question like these are going to be with a lot of potential abbreviations, you can write it out the first time and then abbreviate it after that. Or if you want, at the top or at the bottom of your essay question, you can just have a key, right? Transverse tubule or T-tubule equals transverse tubule, right? You know, SR means sarcoplasmic reticulum or whatever it is. So, I mean, you can just have a key at the bottom or at the top of your essay question too, and then you can do the abbreviations during the essay question. On the lab exam, where, you know, spelling matters, it's important to spell things out and not to abbreviate it. But on an essay question, as long as it's written out once somewhere, you're going to be fine. All right, we have that transverse tubule. We also know that that transverse tubule is going to be associated with, speaking of the sarcoplasmic reticulum, the sarcoplasmic reticulum. We've got this big, it's like a mesh-like structure, remember, but I'm going to just draw it. Oh, I'm going to cheat. Hold on. Let's draw a little bit like that more, expand it out a little bit. Our sarcoplasmic reticulum. And in particular, there is an enlargement of the sarcoplasmic reticulum where it gets close to the transverse tubule. And what do we call that enlargement?
Terminal cisternae.
Excellent. Although technically I just have one here, so terminal cisternae singular. But the reason I put the plural is because we know that there's going to be another sarcoplasmic reticulum with another portion of enlargement on this side as well. And in fact, I'm going to cheat and move my labels over to this side. And these three things together, the two terminal cisternae and the transverse tubule, collectively are called what again?
Triad.
There you go. Excellent, the triad. Oops, no, hold on. That stays there. All righty. We know that this sarcoplasmic reticulum is sitting on top of, let's do it this way, our myofibril. Which is that contractile organelle, which we know is made up of repetitive pattern of structural and functional units. And what is that? A functional and structural unit of the myofibril?
Fascicle.
Close. Remember, fascicle is a bundle of things, but remember that's at the muscle level. We're inside the muscle cell now. So it's the special arrangement of proteins from Z disc to Z disc.
Myosin.
Myosin's in there. We got the myosin, we got the actin, we have all of those, and they're in those repetitive units, right, that are defined by the Z disc. There we go, sarcomeres. And we'll remind ourselves over here of what a simplified version of this looks like. We have a Z disc. Coming off of the Z disc, we have our actin filaments. In between the actin filaments, we have our big thick filament, our myosin, that has its myosin heads. And again, those myosin heads want to bind to the actin all the time, but it's not able to because, let's change the color of it slightly just so that we have that thread-like regulatory protein that is the tropomyosin. And sitting on the tropomyosin are those handle-like proteins, the troponin. There you go. Hopefully all of this sounds vaguely familiar. There really isn't any new information here. This is just a reminder of the things, although it's been a week since we've talked about this stuff, and we've covered a lot of material since then, so it's okay if it's a little bit rusty. But there's really no new information here. But we can add a little something to what we've got here based on what we have before. Of course, over here, since it's not a major part of what we're talking about, I'll go ahead and abbreviate this to remind us that this is our neuromuscular junction over here, made up of the synaptic end bulb and our motor end plate and the synaptic cleft. We know the job of the sarcoplasmic reticulum is to store massive amounts of calcium. As I mentioned, outside of the matrix of our bone, the matrix of your bone has the highest concentration of calcium in the body, but the sarcoplasmic reticulum of your muscle cells has the second highest concentration of calcium in the body. There's a massive, massive amount of it, and it's stored and it's locked up there and it's protected because, as we know, calcium makes cells do wonky things, right? However, moving those regulatory proteins are a wonky thing that we want the cell to do. So not only do we have our sarcoplasmic reticulum that is going to store the calcium, but also when needed, it will release the calcium. And what that means is we need a way to get the calcium out. Well, an easy way to do that is with something we're already familiar with. We can put some channels in the terminal cisternae in the sarcoplasmic reticulum that could allow the calcium out. Now, are we going to want the calcium to come out all the time? No, so we're going to want to have a gated channel, one that we can turn on and turn off, and it's one we happen to be familiar with. It is a voltage-gated calcium channel. Remind me again what all of that means. Are you talking, Amanda? Your box is green, but I don't know if your mic is on.
Something has to happen in order for that to open, that channel to open.
Excellent. Exactly. Exactly. It is a voltage gated. Gated channels have keys that unlock them. And as you guys mentioned, that key is going to be a change in the voltage to a special membrane potential, negative 60 millivolts, as someone pointed out here, I didn't catch the name. Augustina pointed out here, and we call that threshold. So that's the key that unlocks it, that voltage change to threshold. And then of course, once the door is opened, then what is it that passes through this channel? Calcium, exactly. So calcium is going to come out. All right, so there you go. This is our starting point. Well, with one minor exception. We are in the middle of the process here, so it's probably a good reminder to remind ourselves that this is going to begin when we generate a muscle action potential at the motor end plate. Remember, and then it is going to spread. Well, let's just start there. So we know that muscle action potential is a big positive wave. Remember, as we talked about, it is a 100 millivolt change. Big, huge, positive wave. All right. So this is our starting point. Any questions on this before we go through the process?
So it's very similar to the first process. It's just location is now different.
Well, again, remember these two processes had different goals, but is it going to use some of the same type of things? Sure, absolutely. We're going to see that. But remember, this is a continuous process. And so things are going to move, things are going to change. There are going to be some differences. And yes, to answer your question, this is happening in every single muscle cell. When I decide to contract my elbow, when I decide to flex my elbow, all of the muscle cells that are involved in this contraction are all going to go through this process. And that's kind of where we're going to get to. Our goal was to start with a muscle. We have broken that down into a fascicle, then into a muscle cell, then we looked inside the muscle cell. Now we're seeing all the tiny little changes that are occurring in one muscle cell. And once we understand it in one muscle cell, then we can put it back into a fascicle to see how those work. And then we can put it back into a muscle to see how that works. And then we can truly understand how we move our body through space, talking about the different types of contractions and stuff. So that's been our goal. Our goal was to break this down to the bittiest pieces so we can understand how those bitty pieces work, so we can then build back up and understand how our muscles move our body through space. All right. Any other questions on our starting point before we go through this? All right, excellent. So here we have our starting point. As we mentioned, here at the motor end plate, we generate a muscle action potential, that big positive wave. And as we mentioned, is it going to just stay here at the motor end plate? No. It is going to spread down the sarcolemma. So our muscle action potential spreads down the sarcolemma. Right, because the sarcolemma is specialized for the production and the propagation of a muscle action potential. However, as we know, this transverse tubule is an extension of the sarcolemma, and the tubule is filled with interstitial fluid. So that same muscle action potential that's spreading down the sarcolemma, is there anything stopping it from spreading down the transverse tubule? No, and that's exactly what's going to happen. Our muscle action potential spreads down the T-tubule. Again, this is a big positive signal, that big, huge, massive wave. And again, if you're sitting next to a big, huge, massive wave, chances are you're going to get wet. And the same thing happens here. As that big positive signal spreads down the transverse tubule, it makes the triad more positive. And did we have a fancy name for making something more positive?
Polarization?
Yeah, exactly. So our muscle action potential depolarizes the triad, which of course is the transverse tubule and the terminal cisternae of the sarcoplasmic reticulum. This depolarization, being 100 millivolt change, is that going to be able to cause my sarcoplasmic reticulum to reach threshold? Yeah. And when it reaches threshold, what do you think the immediate effect of that is going to be? True, the calcium channel is going to open. Excellent. Our voltage gated calcium channels open, and calcium rushes out of the sarcoplasmic reticulum. Right? Again, our job of our sarcoplasmic reticulum is to store a massive amount of calcium. There's a special protein inside we'll talk about later that helps to store it. And not surprisingly, there are calcium pumps. And these calcium pumps' job is to take any calcium that they find and lock it up in the sarcoplasmic reticulum. Right? One of the cities up here in Northern California with the highest concentration of criminals is Folsom. Does that mean that no one should ever live in Folsom?
No.
No. Why should you live in Folsom, even if there are a ton of criminals there?
Because they're locked up.
Yeah, because they're locked up in the Folsom prison. Absolutely. Right? And as long as they're locked up, they can't cause any problems. However, what happens when there's a jailbreak? And that's exactly what happens here. When these voltage-gated calcium channels open, we get a massive rush of calcium out of the sarcoplasmic reticulum. All this calcium is set free. All right, so now, as a result of this, we get an increase of calcium, not so much in the cell because it was all in the cell to begin with, but an increase in calcium in the cytoplasm. Oops. Or because we're talking about a muscle cell, remember we can call it the sarcoplasm. Now, as I mentioned, there are all these pumps, but are these pumps going to be able to get all of the calcium back into the sarcoplasmic reticulum? No, some will get back into it, but just like we saw before, we have more leaving than these pumps can put back inside. And so as a result of it, there's an increase in calcium in our cytoplasm. This calcium happens to be coming out of the sarcoplasmic reticulum right over the top of our myofibril. Remember, our sarcoplasm drapes around the myofibril. So our calcium binds to the troponin. And when it does that, Troponin undergoes a conformational change. It's another fancy term. Someone remind me again what that means?
Changes shape.
Yeah, it's just a fancy way of saying it changes its shape. And when it changes its shape, it causes a rotation of the tropomyosin. So basically, when it changes shape, it rotates the tropomyosin on the actin. Oops. And notice when it changes its shape and rotates that tropomyosin, now the myosin binding sites, or remember what we call the active sites, on the actin are exposed. And now that those binding sites are exposed, now finally myosin can grab onto the actin and start the contraction. And there you go. We have accomplished our goal. Our goal was to bring together the excitation and the contraction. Go from that big positive electrical signal to the myosin and the actin interacting. And again, we did that by using calcium to move our regulatory proteins out of the way. Well, remember, we saw some of that in the last class. It depends. There are several different types of paralysis. If you think about it, in the last class, we learned about, well, really 3 types of paralysis. One type of paralysis, remember, was where we broke a protein inside of our synaptic end bulb, so we couldn't release acetylcholine, and then the muscle couldn't contract. Right? There was nothing wrong with the muscle cell. If we sprinkled a little acetylcholine on the muscle cell would contract just like normal. We just couldn't voluntarily control it anymore because our synaptic end bulb had been broken. Or remember, we talked about that curare, which is putting the gum in the lock. So even if we dumped a bucket of acetylcholine on the muscle cell, even though the muscle cell wasn't damaged, we wouldn't be able to get the muscle to contract because it couldn't be stimulated by the acetylcholine. Or we talked about how tetanus binds and activates, causing the muscle to be in a constant state of contraction. Right? That's a paralysis as well. I can't control this muscle, not because it's so flaccid I can't do anything with it, but because it's in a constant state of contraction. And so I'm not able to control it. And remember, we called that a rigid paralysis. So those are plenty of examples of how we were able to interfere with this process without even getting inside of the muscle cell. Now, you do have the right idea in that this process can be modified. And we'll talk about that a little bit later, but absolutely, this can be modified as well. Just like we could modify things at the neuromuscular junction, we're going to be able to modify things here. It's a little bit more complicated inside the cell, but we will talk about it and try to make some sense of it. All right, excellent question. Any others? All right, let me get rid of those because I don't want it to confuse us. All right, and take that. All right, Let's go to the pretty pictures from your textbook and do this again. Here, we again see that anatomy that we were just talking about. Notice we have our sarcolemma and our transverse tubule. Next to the transverse tubule, we have the terminal cisternae of the sarcoplasmic reticulum, forming our triad. It is sitting on top of our myofibril. Here we see the myomesin, here we see the myosin and the actin. Oh, and way over here we see a little bit of Z disc, it looks like. And we see some examples of some voltage-gated calcium channels. So remember, initiation of communication, and that's what we're talking about right now. Of communication, of excitation, contraction, coupling starts with our muscle action potential spreading down the sarcolemma. And as it spreads down the sarcolemma, it then spreads down the transverse tubule, spreading across the triad and depolarizing the triad. That big positive signal easily gets it to threshold. Because what was threshold again?
-60.
Yeah, -60 millivolts. We only need a positive 10 millivolt change. And remember, action potential is 100 millivolt change. So it's more than enough to get it to reach threshold. That depolarization opens our voltage-gated calcium channels, and we get our jailbreak. All of that calcium is able to escape from inside of the sarcoplasmic reticulum and come out to interact with our myofibril. Notice here we get an up close look at our myofibril, and notice in its resting state, our troponin is sitting on top of our tropomyosin. And our tropomyosin is blocking the binding sites of our actin. But when the calcium is released, the calcium binds to the troponin, and the troponin changes its shape. It undergoes that conformational change. And when it does that, it rotates the tropomyosin. And now notice all of our binding sites are exposed. And now that they're exposed, our myosin head is finally able to grab onto the actin and start its contraction. Notice all of these pictures are from your textbook. We do have some other pictures that kind of show this process as well. This one's kind of a little bit more similar to the one that I was drawing. Again, we have our neuromuscular junction, our muscle action potential spreading down the transverse tubule, depolarizing our sarcoplasmic reticulum, releasing the calcium, which moves the regulatory proteins and lets myosin bind to the actin. But here I think is my favorite one. Notice for all of these, we've been looking at this in a longitudinal view. What I love about this picture is they're showing it to us in a cross section. So notice here, we have our big, thick myosin filament and one of its myosin heads. Remember, it's a dimer. Here's another myosin and one of its dimer heads. Here's our dimer actin, and here in yellow, we see that binding site that the myosin wants to grab onto. But it can't because that pesky tropomyosin is in the way. And there's troponin, our little handle on top. When the muscle action potential spreads down the transverse tubule and spreads to the sarcoplasmic reticulum, it opens those voltage-gated calcium channels, and calcium is released into the cytosol, into the cytoplasm. where it binds to the troponin. And when it binds to the troponin changes its shape. And as it changes its shape, it rotates the tropomyosin. So now our binding sites are exposed and the myosin heads can grab on. All right. Questions on that? One more. Again, here we see again, the cell is at rest. All the calcium is locked up in the sarcoplasmic reticulum. Here are those pumps that we talked about, keeping it locked up in the sarcoplasmic reticulum. And remember, as I mentioned, there is a special protein found inside of the sarcoplasmic reticulum that helps to store and keep the calcium inside. It is called calsequestrin. Right? If you've ever heard, like in many very high-profile criminal cases, they will sequester the jury, where basically they lock them up in a hotel with no TVs, no newspapers, no Wi-Fi, so they can't get any kind of external information. They're hidden away so they can't be biased by everything that people are saying on TV and in the radio and on the news and newspapers and all that kind of stuff. So they sequester them. Well, calsequestrin sequesters calcium, locks it up inside. So that when our muscle action potential spreads down the transverse tubules, depolarizes the sarcoplasmic reticulum, all that massive amount of calcium can come out, move those regulatory proteins so myosin and actin can interact. Questions on that? All right, excellent. Then let's do two things. Now we can come back to our table here. Now that we've talked about the process, what is the last step of excitation contraction coupling?
Myosin can grab the actin and start the contraction.
Okay, but why?
Because because it starts with calcium, I guess, binding.
True, yeah. So we use the calcium. Remember, our goal was to move the regulatory proteins. So what was the last thing that happened? The troponin rotates the tropomyosin off the active site, or again, off of the myosin binding site. So myosin can grab the actin. And there you go. And guess what? Not only is that going to be the last step of process 2, but guess what else it is?
First step of process 3.
Excellent. And while we're here, what is process 3?
The contractile cycle, meaning it's a continuous process.
Excellent, contractile cycle. Or remember, we could also call it the sliding filament theory. Both of those are acceptable. What is our goal of the contractile cycle?
To allow the myosin and actin to interact with each other.
True. And what does that do when the myosin and the actin interact?
It changes the length of the muscle cells.
True. And it does that because our goal here is to
generate a force.
Yeah. Exactly.
Okay.
We want to use myosin and actin to generate a force, right? To produce a tension. And that is going to move the body through space. And we now know that the first step in that is going to be to rotate that tropomyosin off of the active site so the myosin can grab onto the active. Excellent. All right, we caught up on there. Let's come back to here. Notice up to this point in blue, just like we did last time. Let's move my transverse tubule down here. So it's out of the way. See this there. Here, so far, we did initiation, which again, remember, is the start of the excitation contraction coupling. And now that we've initiated this process, what do we need to do next? Terminate it, exactly. Or end the process. Absolutely. Now, initiation started with a muscle action potential being generated and spreading. But remember, when we terminated communication at the neuromuscular junction, we stopped producing muscle action potentials. And when we're going to terminate it, that's what's going to happen here. We stop producing muscle action potentials. So there's no new muscle action potential, no new big positive signal. When there's no new positive signal, what starts to happen to our cell that was depolarized before?
Repolarizes.
Yeah, it's going to go back to rest. Absolutely. The sarcolemma repolarizes to the resting membrane potential.
So is this still part of process two? Process two.
Yes, this is the termination of process 2.
Okay.
Right? Remember, our
Right.
goal is to first turn the switch on and then turn the switch off. And so that's what we're doing. So we want to be able to contract the muscle. We want to be able to relax the muscle.
I'm just asking just because usually the last step is the first step of the next process, but
And if you think back to when we terminated communication at the neuromuscular junction in the last class, that ended with no new muscle action potentials being produced at the motor end plate, right?
Right.
And that's exactly where this begins. There's no new muscle action potentials at the motor end plate, no new muscle action potentials spreading down the sarcolemma.
Okay.
So it is a continuous process. Yep. All right. Now, with no new action potential, then that means that our triad will repolarize. And if our triad repolarizes, what's going to be the immediate effect of that? There we go. Excellent. With that triad repolarizing, our voltage-gated calcium channels close. And when they close, there's no new calcium into the cytosol. So no new calcium is going into the cytosol. But isn't there still plenty of calcium already in the cytosol? Yeah. So what are we going to do about those?
Calcium pumps.
Yeah, exactly. Now we can again start using that calcium pump to our advantage. Now with no new calcium coming out, our calcium pumps can remove the calcium from the cytosol. And so the calcium levels drop in the cytosol. with less calcium. Oh, let's say it this way, no calcium to bind to the troponin. What's going to happen to the troponin?
And close the active sites.
Yeah, our troponin is going to go back to its original shape. And when it goes back to its original shape, as you mentioned, it is going to rotate the tropomyosin back over the active site. So and once this is rotated back, is everything okay with the dog? When the tropomyosin rotates back over the active site of the actin, then myosin can no longer bind to the actin. And when that happens, our muscle relaxes. And that is how we terminate excitation contraction coupling. Questions on that? All right, excellent. Now, if you remember in the last class, when we were talking about possible essay questions, we definitely mentioned that initiation of excitation contraction coupling and termination of excitation contraction coupling are two essay questions that I guarantee are in that bucket of questions you're going to be randomly assigned. But remember, we said modification wasn't going to be, because modification of this process is a little more tricky. Remember, as we were asked earlier, we talked about how you can get that flaccid or that rigid paralysis here at the neuromuscular junction. Well, we can interfere with this process as well, but interfering with this process really relates more to what we commonly think of as fatigue, right? And we'll talk about fatigue in more depth as we start building back up to it. But basically, the short answer to this, and it isn't a short answer question, is that obviously how much power we produce is going to be determined by the number of myosin heads that grab and pull. So one of the things that happens as we exercise, we break down our oxygen and our glucose to make ATP. But as we continue to work out, we start to build up, among other things, lactic acid inside of the cell. And why might it be important that we, or significant, that we produce lactic acid inside of a cell? Well, do your muscle cells like lactic acid?
No, it lets you know that you're sore.
Right. It's one of the things that leads to the fatigue of the muscle. Absolutely. It is toxic and can be damaging to the muscle. But the other thing about it is it's an acid. And as we produce an acid inside of a cell, we change the pH inside of that cell. And what's something we learned about that is really sensitive to pH changes? What macromolecule did we learn about that is super sensitive to pH changes? Come on, guys, it's a cumulative class. Proteins, absolutely proteins. And what is our muscle cell chock filled with?
Protein.
Proteins,
Proteins.
absolutely. So do you think that change in pH could change the function of this cell?
Yes.
Yeah. In fact, one of the proteins that is super sensitive to pH changes is actually our friend troponin. Oops. Troponin is super sensitive to pH changes. And so as the pH changes a little bit, its binding site changes, and it's harder for calcium to bind to it. So let's think about this. You make the decision to contract the muscle. You send the neural action potential. At the neuromuscular junction, it gets converted into a muscle action potential, spreads down the muscle cell, opens up the voltage-gated calcium channels. Calcium rushes out over the top of our myofibril. But now if I have a hard time getting that calcium to bind to the troponin, am I going to be able to move the tropomyosin as easy? No. And if I can't move the tropomyosin, are as many myosin heads going to be able to grab and pull? No. And if I'm not as able to grab and pull it with as many myosin heads, what's going to happen to the strength of my contraction? It's going to be weaker, absolutely. So just that one tiny change caused by a change in pH suddenly makes my muscle weaker than it was before. So notice this can be modified in this area, but it's a little bit more of a complex process. And it's one of the major factors that we will, after we understand all of this and we start building back up, we will talk in more depth about fatigue. Yes, Augustina, you have a question.
This one's kind of more complicated to type it out. So
Okay,
when
perfect.
someone takes like supplements to basically like decrease the fatigue and exhaustion so you go on for longer. Like I know bodybuilders do this, but they take supplements that purposely make them not fatigued so they keep going and pushing their limits. Is that like what does that do in this like molecular level?
So, great question. The primary cause of fatigue in a muscle is what happens when a muscle is not able to produce energy aerobically. We will talk about this in more depth, but I'll give you the short answer of this here. Our goal is to completely break down our glucose and release as much energy as possible so that the muscle can be as efficient as possible. But to do that, we need oxygen. If we can't get enough oxygen to the muscle cell, or if the muscle cell is more active, then it's using energy faster than the muscle is able to get the oxygen that it needs to completely break it down, then we start to break that glucose down anaerobically, and that's what produces lactic acid. So lactic acid has a big effect on our ability to make ATP and our ability to get oxygen. So most of those supplements that bodybuilders or endurance athletes take is either something that is going to be able to increase the oxygenation of the muscle so we can get more oxygen to it. It's one of the reasons why endurance athletes will blood dope. They'll pull a pint of blood out of their body and then they'll, right before the race, shove that pint of blood back in there so they have more red blood cells so they can carry more oxygen to the muscles, right? Or they'll take a supplement like creatine phosphate, which is one that helps to, it's basically an energy storage molecule that the muscle cell can use to be able to store more phosphates so that it's able to more readily make ATP. So they'll do things like that. And the goal of those types of things are to stave off that anaerobic respiration, stave off that lactic acid so they can continue to use that muscle for longer without fatiguing. Now, like I said, we'll talk about fatigue in more detail later once we've finished building our way back up, but that's kind of the short answer to the question. Was that satisfactory? Excellent. Perfect. All right. So obviously modifying this is a more complicated question. We'll talk about fatigue later, and fatigue is a possible essay question, but not related to these that we guarantee are going to be on the exam. All right. Questions on this? All right, excellent. Well, just that easily then, we are through process 2. So now we can go back to this. And the last thing that we need to do to understand how to contract a muscle is to talk about process 3. And that's what we will do next. However, we will do that after the break. So let's go ahead and take our first break and do that first. Again, it's 9:11 now, so we'll take a 15-minute break. So we will restart at 9:26. And I will start the recording at that time. So any more questions on excitation, contraction, coupling before we take our break? All right, awesome. Then I will see you guys in 15 minutes. All righty. Any questions before we get started?
I do have one question, but it's a little off topic.
All right.
Next semester, is everyone in person? I don't really know what the schedule's like, and I was wondering if you're teaching 431 in person or if it's like a hybrid.
It's a great question. So my caveat to this is that things are still subject to change.
Right.
As of right now, the expectation is most, if not all, classes will be fully on campus because of the vaccine mandates for the district. You know, all the instructors, all the staff, all the students should be vaccinated or should be being tested daily if they've gotten their waivers. And that is the expectation. There is still some desire from some instructors to continue to teach online, either fully or in a hybrid fashion. The problem that we're running into with that is we can get that through curriculum to get the approval of it, but some nursing programs are saying now that we're out of, and not just nursing, but dental programs and things like that, now that we're out of the emergency status, are not willing to take classes that have that don't have on-campus labs. So if you're not, like this class, for instance, doesn't have an on-campus lab, a hands-on lab. So if you're not getting that hands-on lab, they're saying that they're going to stop accepting those classes because they want their students to have that hand-on experience. So because of the reluctance of some programs to accept it, I don't know how viable a fully online class will be moving forward. Some instructors may still try to do hybrid, where you would have an asynchronous lecture and then class on campus, but that is not a format that I'm comfortable with. So I don't like the asynchronous lecture. As you see, I like to interact during my lectures. So my hope is that I will be on campus next semester. So that
Okay, thank you.
I don't know, you know, a tremendous amount can change in the next 3 months. So who knows, but I believe I should have at least one 430 class. Usually the goal is to have it at the same time. So since we're meeting Monday, Wednesday mornings at 8, we should be meeting on campus Monday, Wednesday mornings at 8. If this is the time that works for you, then hopefully that'll be a time that works for you next semester as well. But we have to take all of that with a kernel of salt because you know, things could change dramatically over the next couple of months.
Right. All right. Thank you so
Yep.
much. I appreciate it.
Yeah. All righty. Any other questions? All righty then. So what we're left with then is process 3. We already know that's the contractile cycle or the sliding filament theory. We know its goal to use myosin and actin to generate a force to produce our tension. And we know the first step of that, the movement of the regulatory proteins using calcium to change the shape of troponin to move the tropomyosin so that the myosin and the actin can grab. But once again, just like we did before, to fully understand what's going on, we have to remind ourselves of the anatomy first. So again, process 3. The contractile cycle. And again, our goal is to produce tension. Right, to generate a force. Excellent. And we need to understand our anatomy to do this. The anatomy we're going to care most about is the anatomy of the sarcomere. And again, this is anatomy we're already familiar with. We know that there are 7 proteins you are responsible for that are going to make up the sarcomere. The structural proteins start with our Z disc. And remember, our Z disc is the boundary of a sarcomere. Here, we can cheat a little bit. Move this one here to remind us that this is one sarcomere here, and then there's another sarcomere going off the other side as well. Attached to the sarcomere is going to be our actin. So we'll put an actin this way, and an actin this way, and an actin there, and an actin there. And again, as we mentioned, actin's going to go off this way as well. We know in between those, we have our big thick filament, our myosin. I want these to overlap a little bit more, so let's make this a little longer. And we know that that myosin has heads that are the motors. And someone remind me again what a motor does?
Does it convert chemical energy into mechanical?
Chemical
Exactly.
and mechanical.
Excellent. So we have the tails, which form the thick filament of the myosin, and then we have those heads. And the heads are the motors. Basically, what they do is they bind an ATP, and then from that ATP, they rip one of the phosphates off. And when you rip a phosphate off of that ATP, what do you get as a result of that? ADP, adenosine diphosphate. Absolutely. And so that's what our myosin head does. It's going to take that ATP, rip the phosphate off so that it has ADP and phosphate. And remember, when it does that, it releases a tremendous amount of energy. This ADP, this phosphate, and this energy are all stored in the myosin head, which is ready to do work. And when it is stored in this myosin head, we call this myosin head primed. Primed in this case means that it is ready to do work. And when a myosin head is primed, it is pointing away from the M line. Remember that our sarcomere has those 3 interlocking proteins at the midline, that myosin. And so when that myosin head is primed, it is pointing away from the M line. So notice on this side, our myosin heads will be pointing in this direction. And on the other side, our primed myosin heads will also be pointing away from the M line. And they will be pointing in that direction. So all of our prime myosin heads point away from the M line. Our myosin, our actin, our myosin, our Z disc. We know we have that, I'll draw it here in brown. Remember, we also have that titin protein. It's not going to play a prominent role in this process. And normally we don't draw it. I'll just draw one there to remind us that the titin is there, since we were talking about all of the proteins. But the two proteins I haven't drawn yet that obviously are important is the troponin and the tropomyosin. All right. So we have all of this here, all of these components that we've talked about. And remember also on in your module, under the lab handouts, scroll down to the bottom and wait slowly for it to load. Okay, not the bottom, too far. There we go. That's what we see here. Remember, basically what we drew is basically this model in the classroom. We have the two Z discs with the actin connected to them, the myosin stabilizing the myosin heads. And then here, if we look closely at the actin, we can see that there is a yellow line and green dots on that. And in fact, I think if we scroll up, the next picture, there we go. We can even zoom in a little bit.
Is that considered one sarcomere?
This is considered one. So yes, this would be one sarcomere. Why? Why am I? Is it because it doesn't like that I'm zooming? Yeah. So come on. Yes, you are correct. Our model makers again made a bit of an error here, and notice their myosin heads are pointing in the wrong direction. I think they're trying to indicate that they're pulling at that point, but you're right. For a resting sarcomere, you are correct. Our model maker here has made an error, or again, we don't like to say they made an error. They just took a little bit artistic license. But we can see that the yellow line represents the tropomyosin and the green is the troponin over the top of it. So this is indeed our starting point for our muscle. And notice we see this nicely on the model. I've done an amazing job of drawing it, but notice also here we have the pretty picture from your textbook that shows this as well. Notice here we indeed do have the myosin heads pointing away from the M line on both sides. Our M line down the center, our actin, Z discs, right? Doesn't show the regulatory protein on these, but it also reminds us of the banding. Remember, the portion that contains myosin is the dark band, the A band. The part that doesn't contain myosin is the I band. Part of the A band, myosin and actin overlap, but there is part of the A band that is just myosin, and that is the H band. And then, of course, smack dab in the center, we have the M line. And at the ends of the sarcomere, we have our Z discs. So that is our starting point for our anatomy. All right. Now, what we are going to do, and what your textbook has done a nice job of doing, is I am going to just grab this piece right here and blow it up big so that we can look at it. Your textbook basically does the same thing, although your textbook grabs both pieces. So notice as we look at this here, there's our actin, There's our troponin and our tropomyosin, and here is our myosin. So this is our starting point, our resting point, and that is where we want to start. But to make things a little simpler for myself, we are just going to grab this and look at that. So let's go ahead and do that. Again, our goal here is to describe the contractile cycle. Oops, that's supposed to be a 3. And again, our goal is to produce tension, to generate force. For our starting point, oops. We have our actin and our Z disc. We have our myosin. And we have our myosin head. And as we mentioned, this is a primed myosin head. So remind me again what that means. Excellent, absolutely. So in this case, you guys are absolutely right. That wasn't quite what I was asking the question of, but I like your guys' answer actually better than what I was looking for. Primed, what I was looking for means that it's ready to do work, is what I was thinking. But what you guys say is actually way more accurate, and I like that a lot better. It has already split the ATP into ADP and phosphate, and it has stored the energy ready to do its work. And as we mentioned, when it's ready to do this work, it is going to be pointed away from the M line. All right. Perfect. I think, yeah, we'll make this work. All right. Now, remember, it's ready to do work, but when does the work begin? How do we start this process? How do we start the contractile cycle? Well,
Doesn't
remember,
the calcium have to bind to the troponin?
yeah, exactly. Remember, the end of process 2 is the beginning of process 3. So absolutely, the start is calcium binds to the troponin, changes its shape, and it rotates the tropomyosin off the active site. So that is how this process begins. So let's go ahead and put our tropomyosin there, put our troponin on it, bind a couple calcium to it, and remind us that this has been moved out of the way. Excellent. Once that has been moved out of the way, as we said, that primed myosin head is now going to be able to bind onto the actin. So let's go ahead and draw my actin and my Z disc. And my myosin. Excellent. With that binding site exposed, our myosin can attach to the actin. Right now, of course, this is anatomy and physiology. So can we just simply say that it attaches to the actin? No, of course not, because we know anatomists hate us, so they have to give a fancy term to us. And in this case, the fancy term is we say that it forms a cross bridge. But forming a cross bridge means that it just basically grabs onto the handle. That's all it's saying. It's just a fancy way of saying that it grabs onto the handle.
So if we use an essay question, forms a cross bridge, do you want us to explain what that means?
Sure, yes.
Okay.
That attaches. Again, you don't have to say that it grabs on the handle, but it attaches. That, yeah, we explain that it attaches, because I don't want you to just memorize a fancy term. Make sure you understand what that term means. Now, when it attaches, now it is finally going to be able to use that energy that it has been stored. Again, that energy has been stored in the head, and the myosin head has been waiting to use that energy. And it is going to use that energy to perform, another fancy word, a power stroke. So it's going to perform a power stroke. So what is a power stroke? Well, a power stroke is basically where the myosin head pivots towards the M line. So let's draw this. So our myosin head has pivoted towards the M line. Now, as it's grabbing onto the actin and as it pivots towards the M line, clearly it is going to pull on the actin and pull the actin towards the M line. And since the actin is connected to the Z disc, when the myosin head performs its power stroke, it pulls the Z disc towards the M line. So notice, as the myosin head pulls both the actin and Z disc towards the M line. Now, remember, we're looking at the right side of our sarcomere where this is happening, but the same thing is happening on the left side. On the left side, the myosin head is grabbing and performing a power stroke as well. So if both myosin heads are grabbing and pulling the actin and the Z disc towards the M line, what happens to our Z discs as these power strokes are performed?
They're getting pulled closer
Yeah.
together.
Our Z discs are moved closer together. And as our Z disc is closer together, what happens to the size of our sarcomere?
Make it smaller.
Excellent. It shortens, it gets short. Oh, I like that. Let's go with shorter, even better. Excellent. Remember, this is not just happening in one sarcomere, but it's happening inside the entire myofibril. So when the sarcomeres get shorter, what happens to my myofibril?
They get shorter.
It gets shorter. My myofibril is connected to the muscle cell. So when the myofibril gets shorter, what happens to the muscle cell?
It's shorter.
And that muscle cell is connected to a bunch of by connective tissues to form the muscle itself. So when the muscle cell gets shorter, what happens to the muscle?
It contracts or gets shorter.
Yeah, the muscle gets shorter as well, right? Remember our goal in all of this. Our goal was to generate a force, generate a tension. And when the muscle gets shorter, the muscle pulls on bones, it generates tension, it produces our force. There you go. Mission accomplished. This is what we wanted to do. And here, lo and behold, we have done it. However, if I'm at that McDonald's trying to get to the roof and I grab onto one rung and I pull myself up one step, am I on the roof?
No.
No. Do you think one power stroke of one myosin head is going to be enough to move my body through space?
No.
No. And again, the dead giveaway on this is the name. Contractile cycle. What does cycle mean?
It's continuous.
Repeating.
It is a continuous process that is going to be occurring. All right. So we need to continue this process. We need to get back to our starting point again. Now, there is one more thing we have to talk about that occurs during the power stroke. As we mentioned, during the power stroke, the myosin head uses the energy, right? That's how it's performing the power stroke. But the other thing that happens is that it also releases the ADP and the phosphate during the power stroke. So during the power stroke, this myosin head, that's too big, is going to let go of the ADP and the phosphate during that power stroke. And so because of that, we now have an open binding site. And what can bind to this open binding site?
ATP.
Yeah. Exactly. So when the ATP and the phosphate leave, we now have an open binding site for our ATP. And that is what happens next. So let's get some drawing. There's our actin. Here's our Z disc. Here's our myosin. So with that open binding site, a new ATP, because obviously the old one has been used. So a new ATP binds to the myosin head. The act of its binding changes the shape of the myosin head. And when it changes the shape of the myosin head, our myosin head lets go of the actin. Again, we have a fancy term for this. Instead of forming a cross bridge, we break the cross bridge. But all that is a fancy way of saying is that when the ATP binds, the myosin head lets go of the actin. It's not using the energy for it. It is just letting go. Breaks the cross bridge. And in fact, I'll move this over here. Notice when that occurs, now my actin's binding site is now open, so a new myosin head could be able to grab onto it. But even though we've broken the cross bridge, is this myosin head ready to do work?
No.
No. So what does it need to do to be ready to do work?
Release phosphate.
Exactly. So for it to be ready to do work, it must split the ATP. When it splits the ATP, of course, we get an ADP, we get a phosphate, and we get energy. And when it does that, it revitalizes or reprimes the myosin head. And remind me again what it means to be primed.
To be
Ready
ready.
to work or not supposed
Yeah.
to
To be ready
ADP.
to do work, it has to be tilted so it's pointed away from the M line. And then, of course, if there's calcium to move the regulatory proteins, that prime myosin head will form a cross bridge. Once it forms the cross bridge, that myosin head will perform its power stroke, pulling on the actin, pulling on the Z disc, shortening our sarcomere, shortening our muscle cell, generating that force, moving our body through space, using up its energy and releasing the ADP and phosphate. So then a new ATP can bind, breaking the cross bridge, and then it can split that ATP into ADP, phosphate, and energy, and the process continues. This process occurs at a rate of about 5 cycles per second. So a myosin head grabs and pulls, lets go, and reprimes 5 times in a single second. So again, I keep using the example of climbing a ladder, but that's really what it's like. If I'm going to climb a ladder, I'm going to prime my arm, stick it out so that I'm ready to grab the handhold. As long as the plexiglass is out of the way, I can grab onto that handhold, forming my cross bridge. I can perform my power stroke where I pull down, generating the force, generating the tension, but then I have to let go again so I can prime again and grab another rung and pull and let go and prime and grab and pull and let go and grab and prime and grab and pull and let go. Basically around and around 5 times a second. That's what our myosin heads do. And again, are we reliant on just one myosin head?
No.
No, a single myosin filament has hundreds, if not thousands of heads. And if I'm climbing the ladder with my 2 hands, do I reach with both hands, grab with both hands, pull with both hands, and then let go with both hands at the same time?
No.
No.
No, that's not going to be a very effective way to climb a ladder. So I'm going to alternate when things are going on. So at any one time, one's going to be grabbing, one's going to be pulling, one's going to be releasing, one's going to be priming. And by alternating these, I can grab and pull, and I'm going to make my sarcomere shorter. Indeed, we are a remarkable piece of equipment. I agree wholeheartedly. All right, we've done this on the board. Let's look at the pretty pictures and the pretty words from the class. Notice again, here is our resting point. Our troponin and tropomyosin are blocking the binding sites. Notice our myosin heads are primed. They have split the ATP into ADP and phosphate. They have their energy, and remember we're over here on the right, so they're pointed right away from the M line, ready to do work. And how do we get them to do work? What's the first step?
Release the phosphate and make ADP.
True, we had to do that to prime it, are absolutely right. But while you're sitting here at rest, all those myosin heads are going to be primed, ready to do work. So while you're sitting at rest, all your myosin heads are going to start getting the ATPs they need, priming and getting ready. So when I actually want to contract, what do I need to do to get this muscle going from this relaxed resting state to a contracted state?
Is there calcium?
Yeah, I use the calcium to move the regulatory proteins. Remember, the first thing I need to do is move those regulatory proteins out of the way. Calcium binds to the troponin. Troponin changes its shape and rotates the tropomyosin. And now my binding sites are revealed. And when my binding sites are revealed, what's my myosin head going to do?
Form a cross bridge.
Form a cross bridge. Excellent. That myosin head is going to grab onto the actin. It is going to form a cross bridge. That energized primed myosin head binds to the actin, forming that cross bridge. Once it forms its cross bridge, right? It's in that prime, that cock position away from the M line. What's it going to do next? Perform the power stroke. Absolutely. Those myosin heads are going to pivot. And as they pivot, they release their energy and they pull the actin and the Z disc towards the M line. Right? This is the power stroke. And as we talked about with this power stroke, again, by pulling the actin towards the M line, our Z discs get closer together. And when our Z discs are closer together, our sarcomere gets shorter. When our sarcomere gets shorter, our myofibril gets shorter. When our myofibril gets shorter, our muscle cell gets shorter. When our muscle cell gets shorter, it pulls on the connective tissue of the muscle. Our muscle gets shorter. And as our muscle gets shorter, it pulls on its tendon and it moves our body through space. Right? This is our generation of tension. This is that production of force. This is what moves our body through space. These little proteins grabbing other little proteins and yanking on them. That is how we move our body through space. Now notice also, as we talked about, during the power stroke, it is releasing the ADP and phosphate. So now the binding site in our myosin heads are open. And what's going to bind to that binding site?
And the ATP?
Yeah, we need a new ATP.
Mhm.
Remember, once you grab and pull on the rung, if you're going to continue to climb, you have to let go. And what causes the myosin to let go? The binding of that ATP. Remember, the binding of the ATP gets the myosin head to let go. But remember, it's not ready to do work yet until it splits that ATP into ADP and phosphate. And then it becomes reprimed. Your book's got these great pictures, but it seems to be missing that step. What I like is this picture here. This picture is not from your textbook, but I think it does a great job of showing this process. Notice, again, it's a cycle. We have a primed myosin head ready to grab. It forms its cross bridge. When it forms its cross bridge, it performs its power stroke. After it performs its power stroke, releasing the ADP and phosphate, A new ATP binds to it and it lets go of the actin. But notice it's not ready to do work yet. It has to become reprimed again. And then once it's primed, assuming the regulatory proteins are out of the way, it can bind. Once it binds, it performs its power stroke. Then a new ATP comes in, gets it to let go, and this process continues. Again, each myosin head on average produces about 5 contractile cycles in a second. Great question. ADP and phosphate, remember this is inside the cell. So that ADP and that phosphate is going to go back to the mitochondria, because remember the job of the mitochondria is to make ATP, to use its energy to take that phosphate and stick it back onto the ADP to make ATP again. So those are going to run off to be reused by the cell. Awesome. Now, again, one myosin head pulling, is that going to generate one contractile cycle, going to massive movements of my body?
No.
No, a single contractile cycle only makes a muscle cell about 0.5% shorter. But when you have lots of myosin heads all grabbing and pulling at the same time within many muscle cells within a muscle, then ultimately you get the movement of the body through space. Questions on that? All right, we started this process. How do we end this process? How do we stop the contractile cycle?
It stops until there's no more calcium.
True, you're right. We could run out of calcium. Or what else could we run out of?
Acetylchloride.
ATP, absolutely. ATP and calcium need to be present for this to continue. Although really calcium isn't used up by this process. ATP is used up, but remember our calcium can be locked back up into the sarcoplasmic reticulum. So it isn't so much that we run out of calcium, but there's none left in the cytoplasm. But when there's none left in the cytoplasm, what happens after that? I'm sorry, guys. I need one moment. I have to take this phone call. Sorry. All right. So you've got the right idea. To stop this contractile cycle, basically, we get rid of the calcium in the cytoplasm. And when we get rid of the calcium in the cytoplasm, what happens?
The active sites are not available anymore.
Yeah, exactly. What's going to happen is our troponin rotates the tropomyosin over the binding sites. And once that happens, our myosin heads can no longer grab. And if our myosin heads can't grab the actin, they can't produce any tension anymore, and the muscle relaxes. And that's it. You can see why that doesn't make a good essay question. Anything that can basically be described in one sentence doesn't make a good essay question. And so you can see why, again, our contractile cycle, we'll just have to explain the process and not worry about termination of it. Now, this may not be a process you have been familiar with before, but I'm fairly certain it's something that you guys are aware of. Because as we just finished talking about, this process is going to continue until either all the calcium is put away in the cytoplasm, or the cell runs out of ATP. Of course, does a cell ever really run out of ATP?
No.
No. Although, is there a situation where it might run out of ATP? What's pretty much happened to the cell if it's run out of ATP, if it's not making ATP anymore and it's run out of it?
It's dead.
It's dead. Exactly. When your body dies, it stops making ATP. So let's think about this. You stop making ATP, and as we know, ATP is being used to pump calcium out of the cell or back into the sarcoplasmic reticulum. But without any ATP, ATP can leak out of the sarcoplasmic reticulum. It can move our regulatory proteins. Our myosin head can grab onto the actin, bind to the actin, perform its power stroke. And then what happens? If there's no ATP left in the cell, can that myosin head let go?
No.
No. And now suddenly the myosin head is locked in place. And what happens to the muscle when the myosin heads are locked in place?
Rough paralysis.
Exactly. It would be kind of a rough paralysis. Although if you're dead, we don't call it a rigid paralysis anymore. What do we call it?
Rigor mortis.
Mortis.
Rigor mortis. Exactly. That is exactly what rigor mortis is. Rigor mortis is basically the myosin heads locked into place, unable to let go because there's no ATP available inside the cell. Right? Again, notice the body, the second you die, doesn't instantly go into rigor mortis in the position that you're in. It takes some time for the calcium to leak into the cell's cytoplasm and to cause that, but eventually it'll lock up into rigor mortis. And does the body stay locked in rigor mortis forever?
No.
No, as the body begins to deteriorate, the myosin heads start to break, the actin starts to break, the Z discs start to break. And as the structure starts to break apart, the body goes limp again, right? One of the ways we can tell how long a body has been dead is by this state of decomposition. Is the body entered into rigor mortis yet? Has it come out of rigor mortis yet? Is it in rigor mortis, right? We can kind of get some sense of how long someone's been dead by where they are in this process. Now, is being dead the only time you run out of ATP?
No, maybe fatigue.
Yeah, although we want to be careful with fatigue. We'll talk about fatigue a little bit more in a second. Fatigue, well, we can do that now. We're going to talk about it, I think, in the next class more, but let's talk about it briefly now. Is fatigue when a muscle fails?
No, it's when you use aerobic, like your
Sure, well,
aerobic, like it gets to can't produce enough.
Okay, you got the right idea. So again, World Series started yesterday, is about to start today. I don't know, I wasn't paying attention. It's Texas and Atlanta, so we don't really care. Houston, but you know, one of the things about baseball is you have these pitchers who can throw a ball 100 miles an hour. So the first pitch of the game, 100-mile-an-hour fastball. By the 7th inning, are they still throwing the ball 100 miles an hour?
No.
No, they may only be throwing it 93 miles an hour at that point. Being able to throw a ball 93 miles an hour is hella impressive. But are they able to perform at the level that they were before?
No.
No, because they're fatigued. Fatigue isn't muscle failure. Fatigue is when the muscle can't perform at the level that you want it to. Right? So muscle failure, on the other hand, can occur if you completely deplete your ATP. That is when the body collapses. Probably one of the most famous cases of this happened, I guess it was like 6-7 years ago now during the Olympics. You may remember during the Olympics, during one of the marathons, there was a female marathon runner who basically got to the track. She could see the finish line from where she was at, and she completely collapsed. She entered muscle failure, where she was no longer capable of producing muscle contractions. And they actually had to wheel out an ambulance and scoop her up and throw her inside. And she never had a chance to actually finish the race. right? Because she could see it from where she was, but her muscles failed. So in extreme types of situations, can you get muscle failure? Can you run out of ATP, deplete your resources so much that the muscles absolutely fail? Yes, but muscle failure and muscle fatigue are not the same thing. All right. Questions on this? All right. We have now come back to this here. Process 3. Is there a last step?
Yes.
What would it be?
Relaxation.
Well, okay, true. Or if we were to think about it, we could say that if there was a fourth process in this, muscle relaxation would be the fourth process in this. I was talking specifically about the contractile cycle. Is there really a last step in the contractile cycle? No, absolutely, you guys got the right idea. Since it's a repeating cycle, there's no last sit-up. Because it just continues around and around and around until, then you guys are also correct, until we get to the muscle relaxation. So remember, as we talked about, I can sneak it in down here. As I mentioned, we have a list of which you are going to be responsible for at least 2 essay questions on the exam. And I'm just going to put I here for initiation. Actually, I'll write it out the first times and then do it after that. There we go. So, if you remember, we came up with 7 possible essay questions, of which I guarantee 2 are going to be on the exam. Initiation of communication at the neuromuscular junction. Termination of communication at the neuromuscular junction. We talked about, and we'll put the asterisk by this, ways of modifying communication at the neuromuscular junction. Initiation of excitation contraction coupling. Termination of excitation contraction coupling. Contractile cycle. And as you just mentioned, we can also think of as its own independent step, muscle relaxation. Now, the good news is this last essay question does not have any new information in it. All it is the termination of communication at the neuromuscular junction plus termination of excitation contraction coupling. So let's do that. If you got the essay question on the exam, to describe the process of relaxing a muscle, walk me through it. What's the first step in this process?
To decide to relax when it begins in your brain.
Neural action potential stops.
Yes, absolutely. There you go. First, we make the decision to relax the muscle. And as a result of that, we stop making, or stop sending, let's say it that way. We stop sending, making new neural action potentials. So no new neural action potentials down the axon to the synaptic end bulb.
Repolarization.
Excellent. So this leads to a repolarization of the synaptic end bulb. What's the immediate effect of that?
No new muscle action potential.
Channels.
There you go.
Closed.
The immediate effect is exactly our voltage gated calcium channels close. And when those close, there's no new calcium into the synaptic end bulb. Of course, that's only half of our problem because we still have calcium in there. So what are we going to do about that?
Calcium pumps.
And what happens once the calcium is removed from the synaptic end bulb?
Concentration of it decreases.
Yep.
And
And then the
There's no more-
repolarization.
Troponin
more
blocks the
urge.
binding sites.
Okay, so again, no more calcium in the synaptic end bulb. What was calcium doing in the synaptic end bulb?
Depolarizing.
Well, no, the action potential depolarized it. What did
calcium
actually
was triggering the exocytosis of the
excellent. Calcium was
ACA
causing the release of the exocyte, the acetylcholine. So with no more calcium, no new vesicles of acetylcholine can bind to the plasma membrane. And so no new acetylcholine, I have to spell it out the first time. Oops, that's not any. So no new acetylcholine into the synaptic cleft. However, isn't there already acetylcholine in the synaptic cleft?
yes.
Yeah, so we got to get rid of the acetylcholine in the synaptic cleft. And how many ways do we have to do that?
3.
What are they?
Sodium and potassium.
The
Excellent. Well, here we go. One of us.
acetyl.
So we're going to use the enzyme acetylcholine esterase
And
excellent.
simple diffusion.
diffusion.
And some endocytose back into the
Diffusion away and excellent. So now with no acetylcholine in the synaptic cleft, what happens next?
the
What's
gates
the-
close.
Yeah, what gates?
The chemical cation.
Excellent. Our chemically gated cation channels close. With those closed, no new sodium into the cell. And of course, no new potassium leaves. But was it the leaving of the potassium that really caused the change?
No.
No,
No.
the sodium coming in. So with no new sodium coming in, our motor end plate repolarizes. And when it repolarizes, what happens?
Drops below the threshold and there's
Yep.
no action potential produced.
Excellent. So no new muscle action potentials are produced. So our sarcolemma repolarizes. And what else repolarizes? Excellent. Our transverse tubule repolarizes, which, as you mentioned, repolarizes our triad, which of course is the T-tubule plus the two terminal cisternae. When our triad repolarizes, what happens to our sarcoplasmic reticulum? There we go, excellent. Voltage gated, and I think I wrote voltage gated calcium channel out earlier. Did I spell it out? Yeah, I did. Voltage gated calcium channels on the sarcoplasmic reticulum close. So when those close, there's no new calcium into the cytosol. Or again, cytoplasm or sarcoplasm, whichever term you like. However, don't we still have calcium in the cytosol?
Yes, and that's what we have the calcium pumps for.
Excellent. Calcium pumps remove the calcium from the cytosol. With no calcium, what happens?
There's no calcium to bind to the troponin.
Yeah.
So the troponin returns to the original shape.
Excellent. When it returns to its original shape, what does troponin do?
Well, it rotates the tropomyosin over the active site.
And when the active site is covered,
Myosin can't bind to the actin, so the muscle relaxes.
and there you go. Notice there's no new information there. Those are just the two steps we talked about, right? Step one, termination of communication at the neuromuscular junction, starts here with the decision. And where would it go to? If the essay question on the exam was describe the process of terminating communication at the neuromuscular junction, the first thing you would say is that we decide to relax the muscle. And what would be the last thing you said?
Repolarization of the motor end plate.
Yeah, well, and no new muscle action potential is produced, absolutely. So that would be termination of the neuromuscular junction, of communication at the neuromuscular junction. So purple. If, on the other hand, I ask you to describe the process of termination of excitation contraction coupling, where would you start that essay question? Excellent. We would start that where the last one ended, right? No new muscle action potential is produced. Yeah, let's hold on. Let's do that. Half and half. And then, of course, where would it stop?
The active site is covered and the muscle relaxes.
Right. Which is why termination of the contractile cycle isn't a good essay question, because basically it would just be active site of actin is covered and the muscle relaxes. So that's why we don't have the third step as part of the of the relaxation, because it really, once it's the binding site is covered, your cycle stops. So there you go. Right here, you have the answers to 3 of your 7 essay questions that could be on your exam. Questions on that?
Would you need it to be more descriptive than that, or is that?
I don't think we could be more descriptive than we did right here. I purposely was extra descriptive in this when we went through this, right?
Okay.
So yeah, I don't think it's possible to be more descriptive than this. And again, yes, it is a bit lengthy, but these are the questions that are going to be worth 8, 9, 10 points on an exam. And remember, the easy way to think about what you're doing is, again, think of what is the immediate effect, right? Again, we could describe muscle relaxation in we decide to relax the muscle, we stop producing a neural action potential, our muscle cell stops producing a muscle action potential, and the muscle is going to relax. That statement is 100% correct, but it lacks all the detail. So a good trick to help you to be successful in having the detail needed is to, again, think what is the immediate effect, right? Notice when you stop producing a neural action potential, the synaptic end bulb stops releasing acetylcholine, but that's not the immediate effect. The immediate effect is that the synaptic end bulb repolarizes. The immediate effect is that the voltage-gated calcium channels close. The immediate effect is the pumps get rid of calcium. And with the calcium gone, we stop releasing acetylcholine. So when you're writing these essay questions, think in terms of what is the immediate effect, and that'll help you to get all the details. All right. Questions on that? This took a little longer than I hoped, but I think it was important to take the time to go over this and make sure we understand this. So let's go ahead and take our last break. And then when we come back, we will switch gears into the origins, insertions, and actions. So any questions on this? All right. The good news is now that we understand how all the bits work, starting, what day is today? Today's Wednesday. Starting on Monday, we get to start to build back up. Now we're going to look at what's happening in an entire organelle of the myofibril, and then in a muscle cell. And then we're going to put the muscle cells together into groups, and we're going to slowly start building back up again. All right. Awesome. Excellent. So that is that. So let's go ahead. I'll go ahead and leave this on the board for the break. Let's go ahead and take our last break. It's 10:37 right now. So we'll come back at, I guess that'll be 10:52. And again, have your, have your handouts, have your study guide, have your blue and red pens, blue and red pens, and I will see you back here in 15 minutes. Any questions before we get started? All right, looking at the slides, I realized there was one last thing that I wanted to show you. If you've had the opportunity to go into the classroom, and hopefully you have, you've seen a lot of great models and other materials in the classroom that have these muscles on them. And remember last time we left off with the platysma, this big, huge draping muscle. If you look at the torsos, if you look at all the head models there in the class, none of them show the platysma on it, because as you can see, it's a big, broad, superficial muscle that covers most of the deep muscles of the head and of the neck and the jaw. However, in the classroom, there is a muscle chart. And notice on this muscle chart, we can see really nicely that big, broad, flat platysma. So this, again, the advantage of being online is that we can use illustrations, we can use materials like that, and you're definitely responsible for the models and stuff in the classroom, but in the chart, So this chart, if we were in the classroom taking the exam, would be the one place you'd be able to see that platysma in the classroom. But luckily we have other illustrations and stuff we can use for this exam. All righty, we are done with the muscles that move the head and neck, and we are on to the muscles that move the trunk and the shoulder. And that brings us to our trapezius. Notice the trapezius muscle is a broad, superficial, superior, and mostly posterior muscle. Notice we see that really nicely with its origin. Let's go ahead and bring up our handouts, and in particular, this one. Excellent. What is the origin of our trapezius?
The occipital bone.
Okay, and not just the occipital bone, but remember that bump on the back of the head, that external occipital protuberance.
Right.
What else?
The spines of C7 and all of the thoracic vertebrae.
All right, what's so special about the spine of C7 again?
It's the most pointiest.
Yeah, it's the
Has the longest sinus process.
Yeah.
and it's known as the vertebral prominence, right? So that vertebral prominence from your 30-point review, that is the spine of C7. And then all of the spinous processes of T1 through T12. Excellent. Notice it doesn't connect directly to any of the rest of the cervical vertebrae. And why might that be beneficial? Why wouldn't you want this big, huge, massive muscle attached to all these cervical vertebrae?
You guys need to be pretty stiff.
Yeah,
It would be a lack
exactly.
of movement.
Yeah, it would reduce the movement. However, we do want a continuous insertion. So what actually happens is that we actually have a big, huge elastic ligament located in the back of the neck region, which, if you remember from when we were talking about the elastic connective tissues, we called the ligamentum nocha, because this is the nuchal region of the body. And so it attaches to that as well. It doesn't attach directly to the bones, but there is an elastic ligament over the top of those that allows for this big, long, continuous origin. And again, notice it is big, and it is superficial, and it is superior in its location. But what is its origin? What's its insertion?
Along the acromion, the spine of the scapula.
So here's the acromion, here's the spine of the scapula. And what else?
The clavicle?
Yeah, the lateral third of the clavicle. Notice this lateral third of the clavicle is actually on the anterior part. It comes up here onto the anterior part of the body. So that's why we say this is a mostly posterior muscle. Notice if we go back to the pictures here, from this posterior view, we can see how the fascicles are traveling around and up. We can see where it gets its name, the trapezius, because again, it's got this kind of trapezoid shape to it, where the fascicles are coming up, big, huge convergent muscle. But we can see how some of these fascicles are going towards the front. Notice if we cheat and we go back to this picture, we can see the teeniest bit of it peeking up over the top right here. But remember, the other way we're going to be responsible for muscles are on these big bodybuilders. And notice on this big bodybuilder, especially with him hunching forward, as they bulk up these muscles, we can very clearly see how some of that trapezius, which we can see right here, his trapezius muscle, is coming forward onto the anterior part of the body. So it is a mostly posterior muscle that comes a little bit onto the front. All right. Questions on its origins and its insertions? All right. So what are its actions? How many actions does it have?
4.
4. What are they?
To raise or
Raise,
elevate.
yeah.
Okay. Elevate the shoulder. Bring the scapula up. What else?
Rotate.
Back the scapula.
Retract, bring it back. And remember, when we bring it back, that also means to abduct. And actually, let's go back to the picture for this. So one of the things that it can do to the shoulder is it can elevate the scapula, bringing the scapula up. It can retract, which remember also means bring it towards the back. So that abduct, bring it back. And you guys said rotate as well. Let's take a look at this. Notice if I were to bring my arm out to the side, and what would that action be called again?
Adduction.
Yeah, abduction.
Or
Abduction, yeah, bringing it away, abduction. Notice if I were to do that and bring my humerus out, eventually there would reach a point where the neck of my humerus would bump up against my acromion, right about when I got here. Does that mean I can't abduct my arm beyond this point?
No.
No, clearly I can. However, the reason I'm able to abduct it more than this is because of what the trapezius is able to do. The trapezius is actually able to rotate the scapula. So basically, you would take the scapula and turn it, rotate it upward so that the arm can then abduct above this point. This is actually something hard to do on yourself, but if there's somebody else in your household, put your hand on their scapula and have them abduct the arm. And as you'll notice, as they first come up, the scapula doesn't move at all. But then as you continue to go up, the scapula will actually rotate. And so you can actually feel that occurring. Notice again, the appropriate terms are to elevate, right? But we can cheat. We know that elevate also means to raise. Retract, which we also know means to abduct. And again, I emphasize the B so we know what we're hearing here. And also rotate. All of these effects on the scapula. A good mnemonic to help you to remember these are to remember the 3 R's. Right? Back in ancient times, and by ancient times, I mean when I was in elementary school. Anybody know what the 3 R's are that I learned in elementary school?
Reading, writing,
And arithmetic.
arithmetic.
There you go. Reading, writing. Does writing begin with an R?
No.
Is arithmetic even a word?
No.
No, it tells you something about the education that I got growing up. You know, we got it, us old people got, but yes. So technically 2 of the 3 things that were R's aren't actually R's, but that's what we learned. My kids, on the other hand, learned a different set of 3 R's. What are the 3 R's that my kids learned in elementary school? Notice the color I used. Close. I was thinking more something a little green, the 3 R's of greenness that we've learned.
Raise, retract, rotate.
No, that's what you're learning in A and P. What my kids learned in elementary school was, there you go, reduce, reuse, recycle, right? So that's what many of you, and my kids included, learned in elementary school, but you are absolutely correct. Here in anatomy and physiology, the 3 R's of the scapula are to raise, retract, and rotate. Of course, there is a 4th action. What is the 4th action?
Extend head.
Extend the head. Remember, we have all this broad converging fascicles coming together. Notice if I just use my superior fascicles, I can bring my head back. I can extend my head. So extend the head, raise, retract, and rotate the scapula. The mnemonic that I always use to remember this for myself, for the trapezius, is to think in terms of if you were making a serve in tennis. If you think about making a serve in tennis, you're going to throw the ball up in the air. And when you do that, you have to have your tennis racket ready. To have your tennis racket ready, you have to bring your shoulder up. You have to bring your shoulder back. You have to rotate your scapula so that you're up here with the tennis racket. And then when you throw the ball up in the air, what do you have to do? If you're going to serve the ball, when you throw the ball up in the air, what do you have to do? Look up to see it. Exactly. So raise, retract, rotate the scapula, extend the head. I'm now ready for my tennis serve. So there you go. Origin, insertion, action of the trapezius. Questions on that? All right, excellent. Let's look at another interesting muscle, and that is the serratus anterior. Notice the serratus anterior. What is the origin of the serratus anterior?
The ribs.
Which ones? Are you
1
just going to
through
say the
8.
ribs on the exam? No. Exactly. You are going to say the ribs 1 through 8. So notice, whoops. We see these muscles that attach. And notice you can see where it gets its name. On the anterior part of the body, we have this kind of scalloped appearance to it where these muscles attach. That scalloped or serrated edge, like a serrated knife, is where we get those, the name for this. It has that serrated appearance to it. So for instance, if we go to our pictures, oops. I know this isn't the right picture, but it's the one that I want, so this will work. Our origin is again on the anterior surface of ribs 1 through 8. 2, 3, 4, 5, 6, 7, and 8. And it lays across the rib cage. Now, what's interesting about this is how it inserts. Let's actually change our view. If we were to change our view on this, to look at it from a superior view. So here is my vertebrae. Let's give my vertebrae its transverse processes and its spinous process and its body. We have all of that. And we'll even give this a sternum here in the front. We know that the origin of the serratus anterior is on the anterior surface of the ribs. Where does it insert?
The anterior surface of the medial border of the scapula.
Excellent. Notice here are my 2 scapulas. And the insertion for this muscle is actually right here on the anterior side of the scapula on the medial border. So notice it has an anterior origin and an anterior insertion. And so what this muscle does is it basically wraps around the ribs, goes between the ribs and the scapula, and then attaches to the medial border of the scapula. Notice if we go back to our drawing where we're drawing the origins and insertions, this could be a little tricky. So again, do this however it makes sense to you. If you just want to draw it on the medial most part of the scapula, that's fine there. If you want to, with a dotted line or something, remind yourself that it is going to be in that posterior part behind the ribs, something that way, or shade it or whatever, or do what I just did. Right here up at the top, draw your ribs, draw your scapula, and then you can draw your insertion right there on the medial border of the scapula. So again, you're not turning this in, so there's no wrong answer. Just draw it how it makes sense to you. All right. But notice when this muscle contracts, like when all muscles contract, it is going to pull the insertion towards the origin. So what effect does that actually have on the scapula?
Protract.
Yeah, this is going to protract the scapula. Now, as we know, when we protract the scapula, those pesky ribs are in the way. So when we're protracting it, it's really going to be bringing it out to the side. So not only could we say that it protracts the scapula, but what other term could we use for it as well?
Abduct.
Abducts, exactly. And so that is bringing it forward, right? And again, if you think about, right, you have that friend you haven't seen in a million years and you go to give them that big hug, or if you're hugging yourself, right, to help to, get rid of your stress cycles, right? That coming and bringing the arm forward, you're bringing the scapula forward, but it's also coming around the edges as well. But remember, we also want to have tension in our muscles most of the time. And when there's tension in this muscle, the other thing that the scapula does is help to hold the scapula against the ribs. This helps to stabilize the shoulder. Sometimes when a person falls and lands on their shoulder and crushes it inside, or sometimes during birthing is a very stressful process for both mom and baby. And as the baby is coming out heads first, sometimes the stretching of the neck, this can cause problems to a cluster of nerves in here known as the brachial plexus. And this brachial plexus is part of what's responsible for holding the scapula in place. And so what can actually happen is if that nerve gets damaged, the scapula will actually wing off. Where basically what will happen with the scapula is the scapula will basically, as it loosens, it's going to, whoops, the medial border comes off of the wall of the ribs. It's what we call this winging of the scapula. And if you get a winging of the scapula, is that going to affect your ability to move and function with your shoulder?
Yeah.
Yes.
Yeah, absolutely.
Can
So
that
that-
happen with whiplash as well?
Yes, it can. Anything that damages the brachial plexus has or injures or traumatizes the brachial plexus has the ability to cause this type of disruption in function. Absolutely. And so that ability to hold the scapula against the wall of the ribs is a second important function. In fact, if we squeeze this hard enough, we can actually squeeze the thoracic cavity. And why would we want to squeeze the thoracic cavity?
To help exhale
Yeah.
for cough.
And when you squeeze that ribs, this is a muscle that helps us to exhale. So when you're blowing out your birthday candles, you squeeze in the thoracic cavity to force that air out. So this can actually is an assisting muscle for exhalation. It's not the primary muscle for exhalation, but it can help in exhalation. Excellent. Now, one more thing I want you to see about this serratus anterior. Notice to see the entire serratus anterior, they had to cut away this big superficial pectoralis major because the pectoralis major covers most, but not all of the bellies of the serratus anterior. So notice again, we can see a couple of those bellies sticking out underneath. Now, this is just something we can see on this illustration. We couldn't actually see that on a person, could we?
Yeah, good.
Yeah.
Builder.
Yeah. Notice here on our chart, under the pectoralis muscle, we can see some of those serratus anteriors. And notice it's much more obvious when the arm is up, like here. Notice with the arm up, we can actually see those bellies of the serratus anterior on our bodybuilder. So it is a mostly deep muscle, but there is enough of it that is superficial that this would be a muscle we would be responsible for identifying on our bodybuilder. Remember, I said it was mostly superficial muscles, but there were a couple exceptions. This is one of those exceptions because a portion of it is superficial and it's super obvious. All right. Questions on that? Do I have that again? I do. Okay, perfect. All right, that is the serratus anterior. Let's talk about the rhomboids. Rhomboid is a muscle group made up of the, actually, this isn't the picture for this, is this? Well, it's one of these. Made up of two muscles, the rhomboid major and the rhomboid minor. Do we care?
No.
Nope. We just need to know them collectively as a muscle group. What is the origin of the rhomboids muscle group?
The
Spinous
spinal process of C7, T5.
C7, T1, T2, T3, T4, and T5. And what is its insertion?
The posterior medial border of the scapula.
Exactly, the posterior medial border of the scapula. Notice we can clearly see this from this posterior view, but remember we just finished looking at this view here with the serratus anterior. Notice with the serratus anterior, another important muscle, and I'll change the colors here. We'll do dark red. Here is the origin on the spinous process for our rhomboids. And its insertion is on the posterior medial border of the scapula. So notice this muscle also has the ability to move the scapula, but in this case, it's going to be bringing the scapula back or bringing the scapula towards the midline. So what kind of action would that be?
Adduction,
Adduct,
AD, and
Or bringing it back, which would
retracting.
retract.
be Exactly. Notice again, you see that long lost friend you haven't seen in 2 years because of COVID, you're giving a big hug, and then you remember they owe you $100. So you bring back your arm to punch them. And think about it. If you brought back both arms to punch them at the same time, which would be a silly thing to do, but let's say instead you're rowing. If you bring both arms back at the same time using your rhomboids, along your spine, you can actually feel the bunching up of your muscles on the midline. So you can see that not only is it bringing the scapulas back, but it's bringing them towards the midline. And so you can feel the squishing of those rhomboid muscles in between your shoulder blades as you're bringing it back. All right. Questions on that? And of course, here's the pretty picture. Notice, again, this is a deep muscle. It is completely covered by the trapezius. So in a bodybuilder, no matter how big these get, we're not going to see them because it sticks out. Maybe a little bit it'll stick out down here, but we don't have to worry about that. We're not going to, it's a deep muscle. You're not responsible for it. It's covered by the trapezius. All right. Next on our list is a muscle named the Pectoralis Minor. Now, anytime you have a minor something, guess what else you have?
Major.
Major, but I don't see Pectoralis Major on this page. Does that mean you're not responsible for the Pectoralis Major?
No.
No, you're going to be. So then why isn't it here?
Because
Maybe you
it's
can't see it.
superficial.
Say again?
It's superficial.
True, it is superficial, but also remember, we're talking about muscles that move the trunk or the shoulder. Our pectoralis major moves our arm. And we'll see, even though obviously the pectoralis major is bigger and it is superficial, and the pectoralis minor is smaller and underneath, it is the origins and the insertions of these that tell us what they do. So it isn't so much their location as it is their attachment points. And let's do that. Let's take a look at where we would find our pectoralis minor. Is this the picture for it right here? Perfect. What is the origin of the pectoralis minor?
The anterior surfaces of ribs 3 through 5.
2, 3, 4, 5. And what is its insertion?
Coracoid process of the scapula.
Coracoid process of the scapula. Excellent. Here we see it in our drawing. And if we go back to the picture, here we see it here. Notice here we see those bellies connecting to the 3 ribs and inserting into that coracoid, that bird's beak that stuck out on our scapula. All right. Questions on that? So let's draw this again. There's our insertion. Here's our origin, 3, 4, and 5. And as we know, when a muscle contracts, we are going to bring, oh, way too big. We are going to bring the insertion towards the origin. So this muscle is going to bring the scapula down. What action would that be?
Depression.
Depressing the scapula. Notice it's going to bring the scapula towards the front. What action would that be?
Traction.
Protraction or abduction, because remember the back is going away from the midline as we're bringing it forward, wrapping around the ribs. But remember, many of the muscles associated with the ribs can help us in our breathing process if we reverse their actions. So notice if I stabilize my scapula with other muscles, then when this muscle contracts, instead of my scapula coming down, my ribs would come up. right? Elevating the ribs for like helping in that inhalation.
There's 3 actions.
So there are indeed 3 actions for this muscle. Has anybody come up with a good activity that's going to help us to remember these 3 actions of the pectoralis minor? It's okay if you haven't. I've had trouble with this one as well. My sister tells me that she uses this muscle all the time to get out of speeding tickets. But I was pulled over just the other day and I depressed my scapulus, I protracted my scapulus, and I elevated my rib cage, and I still got the ticket. So I don't know what she's doing that I'm not that gets her out of it. But if she says it works, we'll maybe go with that. All right. Questions on that? Oh, excellent. I think I hurt myself doing that. All right. So, all righty. That is our pectoralis minor. Let's talk about some other muscles of the associated with the trunk. We've been talking about all these muscles that can help with breathing, but the primary muscle involved in breathing is our diaphragm. What makes the world go round?
Pressure.
Pressure, absolutely, excellent. And this is the perfect example of where we see this. Here is our ventral body cavity. And of course, as we know, our ventral body cavity is divided anatomically by this bell-shaped curved muscle that is the diaphragm. Now, what happens to muscles when muscles contract?
They shorten.
We shorten.
Yeah. And when a curved muscle gets shorter, what happens to it?
Curls in.
Well, it flattens out. You got the right
Flattens,
idea.
yeah.
So when our diaphragm contracts, what ends up happening is that it flattens out. And notice when it flattens out, what happens to the volume in our thoracic cavity?
It increases.
And when our volume increases, what happens to our pressure?
It increases.
Well, so again, let's think about this. Pressure, when we're dealing with gases, or really when we're dealing with everything, but gases in particular, which we're talking about, for instance, in our lungs. And again, I have to work on my analogies because I don't have good ones when we're all sitting at home, but let's pretend we were all in the classroom. All 20 of us were sitting in the classroom. And what I would do is I would blindfold you and I would let you walk around the classroom. And being blindfolded, every once in a while, one of you would bump into a wall. And what I would do is I would count the number of times you guys bumped into the wall. And that would basically be your pressure in that space. Now, what if I instead took all of you and put you in my walk-in closet? You know, even though I have community college kind of scratch, right? Is my walk-in closet as big as the classroom is?
No.
No.
No, of course not. So when you're in a smaller space, what's going to happen to the amount of time that you guys bump into the walls?
It's going to happen a lot.
It's going to happen a lot more. If instead I let you guys roam free on campus and counted the number of times you bumped into the border of the campus, what would happen to the number of times you would bump into the border there?
A decrease.
It would decrease. So when my volume in my thoracic cavity goes up, what happens to pressure?
It decreases.
Yeah, pressure increases. When volume goes down, when volume goes up, our pressure decreases. Yeah, so our pressure decreases. We have a low pressure inside of our lungs, and because our lungs are open to the outside world, There's our throat. There's our mouth. When the pressure in here decreases, air moves in. And what do we call that process?
Inhaling.
Yeah, inhalation. Now, notice to do this, we contracted the muscle. And as we now know, contracting a muscle uses ATP. So this is an active process. But do we leave that diaphragm contracted forever?
No.
No. Eventually, we're going to relax it. And when we relax it, what happens to the muscle?
Muscle relaxes.
The muscle relaxes and it goes back to its original shape. When it goes back to the original shape, what happens to the volume of our thoracic cavity?
Decreases.
Is going to decrease, right? It gets smaller. And when the volume decreases, what happens to the pressure?
It increases.
It increases. And is that air going to want to stay inside of this high pressure environment?
No.
No. And luckily, our lungs are open to the outside world. And so our air leaves. And what do we call that process?
Exhaling.
Exhaling. Now notice for the exhalation, in this process, the muscle is relaxing. If it's relaxing, are we using ATP? No. And so this is a passive process, right? Instead, it relies on the elasticity of the muscle to go back to its original shape. So when we breathe in, it's an active process. When we breathe out, it's pretty much a passive process. Now, blowing out while you're sitting here in class, right? Your chest is not heaving as a result of it. You're just sitting here quietly, and you're basically, your diaphragm is the primary muscle moving air into and out of our lungs. However, are there muscles we've talked about that can bring the ribs up to help us take a bigger inhalation?
Yeah.
Or muscles we can contract to actively force air out?
Yeah.
Absolutely. So while this is the primary muscle for breathing, we do have accessory muscles for inhalation. And what muscles that we've talked about already in this class would help you to bring the ribs up to help in that inhalation? I can
The
think,
pectoralis minor.
okay. I can think of at least two more we've already talked about.
Sternocleidomastoid.
And
The scalene.
scalenes. Excellent. All of those are muscles that are going to help in inhalation. There's one more we haven't talked about yet, so we do have to add one more to this list. We'll get to that in a second. And there are also some accessory muscles for exhalation. And what one muscle have we talked about so far that can help us to compress the ribs and force that air out? We just talked about it today. Squeezes and stabilizes the ribs or the scapula.
The trapezius.
The serratus anterior. There are more, but we haven't talked about them yet. So we'll add those to our list later. But while you're sitting here calmly in class, or laying in bed, just taking that normal resting breath. That normal resting breath is primarily using the diaphragm. And notice that with that normal resting breath, you are only really using ATP half the time, just to contract the diaphragm to bring the air in. And then just the exhale is just passive. This is significant because remember that passive movement of the air out is due to the elasticity of the lungs, the elasticity of the diaphragm, the elasticity of the rib cage. And what happens to our elasticity as we age?
It becomes less elastic.
Yeah, it becomes less elastic. It decreases, absolutely. One of the things that happens as we get older is we start to lose that elasticity to the thoracic cavity and the muscles. So now for grandpa, while he's sitting there, not only does he have to use ATP to bring in his normal breath, but now he has to use ATP to contract those muscles to breathe the air out, right? Sometimes grandpa just looks exhausted, just sitting there, sitting in the chair and breathing. And that's because he is exhausted from just sitting there in the chair and breathing, right? Because now he's having to use twice as much ATP just to move air into and out of his lungs. And so just the act of breathing can be exhausting, far harder than it is for us when we're younger. All right, how are we on time? Doing good, excellent. Questions on that? All right. This is our diaphragm. Again, here we have this nice inferior view of it that we can see. Again, it's that anatomical boundary between the thoracic cavity and the abdominal pelvic cavity. So we see there has to be some room for blood vessels, nerves, even the esophagus to sneak through that. But technically our diaphragm isn't the only diaphragm. We also have what is called the urogenital diaphragm. And with the urogenital diaphragm, the primary muscle of the urogenital diaphragm is a muscle known as the levator ani muscle. Levator ani muscle is a muscle group made up of the pubococcygeus and the iliococcygeus. And while pubococcygeus and iliococcygeus are incredibly fun to say and to spell, are you actually responsible for them?
No.
No, you just need to know them collectively as the levator ani. Notice here in our female illustration, we can see the majority of this levator ani muscle. It goes forward a little bit. Notice, again, it's deep in here, so we can't quite see the end of it. But notice it helps to support our organs in our pelvic region. It also plays a role in helping to assist in defecation or controlling urine flow. It also plays an important role of providing support for the vaginal canal during birth. Obviously, males have the exact same thing as well. And here we see the boy version of this. Again, here we see that levator ani muscle, and again, it goes forward, and this plays a role in defecation, plays a role in controlling urine flow. Clearly, males are not passing anything baby-wise through any holes here, but females are. And so again, the job of this is to provide that support. Like I said, you elevate it during defecation to help in the assisting. If you were ever urinating and you needed to stop urinating midstream, right? You were at that bar, you had to urinate, the line is 75 deep for the restroom, and there's these perfectly good bushes outside. So there you are outside, urinating, avoiding urine from your body, and the police spotlight hits you. Do you keep urinating at that point?
No.
Probably
No,
not.
you make that contraction that stops the urine flow. That contraction that you make to stop the urine flow is basically the contraction of that levator ani muscle. I make a point of emphasizing this because not every woman has had given birth before, but clearly passing a basketball through this vaginal canal can be an incredibly traumatizing experience to the female's body. And it can weaken this urogenital diaphragm. You often hear examples of a female shortly after giving birth. If she laughs too hard or coughs or sneezes, what can happen in those instances as a result of that? Coughing, sneezing, or laughing?
Accidentally pee.
Yeah, they may leak a little bit. Why do they leak a little bit? Because of the weakening of the levator ani muscle. Now, tinkling a little bit when you laugh isn't that big of a crime. However, if this muscle is weakened enough, you can actually get a prolapse of the vaginal canal, where the vaginal canal will actually descend out of the urogenital diaphragm and actually has to be put back in there surgically. And that is nothing to be funny about that. So one of the things that pregnant females are encouraged to do is a set of exercises known as
Kegels.
Kegel exercises. Absolutely. Those Kegel exercises are basically doing that activity that I just recommended for you. Contract that muscle like you were stopping during urine flow. You make that contraction, you hold it for a count of 10, you relax, you do them in sets of 10. And the advantage for that pregnant woman is you can do them all the time. While you're sitting in a red light, you can do 10 of them. While you're standing in line in Starbucks, you can do 10 of them. I've just done 7 while I was sitting here right now, and you have no idea, right? Again, obviously, it's not some massive leg movement. You're just making that contraction. And it can help to strengthen that pelvic floor for the female. Now, does that mean that their males don't have to bother doing this? Well, clearly, the male isn't going to undergo the trauma of giving birth that a female will. However, I will point out for those males that are here in the class that some studies have shown that doing Kegel exercises can help to prolong sexual activity, increase sexual endurance. How about we say it that way? So there are benefits for everybody to do those Kegel exercises. All right. And I just did 4 more. And I'm sure a couple of you did too. So I know nobody wants to admit it, but when I talk about them, everybody always does it. All right. Excellent. Questions on that?
I had a question regarding the order. We skipped a few. Was that on purpose?
Yes,
Okay.
because we were talking about the 2 diaphragms. So we were
Okay.
talking about that. So the levator ani muscle. What we skipped were the intercostals. Is that correct?
Yes.
I wanted to do the 2 diaphragms while we were talking about those at the same time. And then now we can talk about the intercostals, because the intercostals is going to require another picture. Notice, as the name indicates, intercostal means between the ribs. And sure enough, that's where you find these. These muscles are located between the ribs. So let's switch to our whiteboard. Get a new spot and draw some new pictures. Here, of course, is our sternum. No. Why not? Here's our sternum. Here are our ribs. And again, obviously, I'm drawing very, very basic examples of these things. Closer. And as we know, there is cartilage that connects the ribs to the sternum. All right. Now, we have 2 sets of intercostal muscles. How did we define them again?
Internal
External and
and external. Yeah.
Excellent. We have an external intercostal and we have an internal intercostal. What do you think the most obvious way these two things are different from each other?
one's external, one's internal.
There you go. See how simple that is? One is internal and one is external. Excellent. But let's use anatomical terms. How else would we identify that? If you were internal, what would you be?
Endo.
Deeper.
Deep.
Anterior?
Exactly.
Oh,
So
ooh.
obviously this is the deep muscle. Whoops. And this one over here, the external intercostal, then would then be what?
More superficial.
So that is a super easy, obvious way we can tell them apart. One is going to be superficial and one is going to be deep. But there are some other big differences between these two. Our external intercostals are indeed superficial, but they are also more lateral. They actually don't get to the cartilage. Of the ribs. And with our external intercostals, the fascicles of the muscle project down and in. So if we were to draw these, starts lateral, its fascicles come down and in. And let's erase that last one, just so that there's no confusion. Yeah, we'll use straight lines so that they're prettier. Now, again, notice I've done this on the right side. These are obviously found on the left side as well. And again, they are lateral, they are superficial, and their fascicles project down and in. On the other hand, our internal intercostals are, of course, deep, but they're also more medial. They actually do start at the sternum and the cartilage. And their fascicles project down and out. So their fascicles start here at the midline. And project down and out. And since they're deep, would we be able to see all of them?
No.
No. And again, that's going to happen on both sides, projecting down and out. There you go. Again, I've done a truly amazing job of drawing this, but let's look closely at the picture. Notice here, when we look closely at our picture, we can see here are the muscles that are deeper. They start at the sternum, they cover the cartilage, and their fascicles are going down and out. Whereas here, we can see these are superficial, they're more lateral, and their fascicles are coming down and in. So we can see this really nicely between the ribs. However, what's awesome about your textbook is your textbook actually does a nice job of peeling the two layers apart for us. Now, obviously, if we're looking at a picture like this, can we tell which ones are superficial and which ones are deep?
Yeah.
Yeah.
By the way they're
Really?
going.
And just ignore the words from looking at the
Oh.
pictures. Just looking at the pictures of the muscles by themselves. Can you tell which ones are deep and which ones are superficial?
No.
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 But can you see the other characteristics that are going to help you to tell them apart?
Yes.
Yes. Notice again, our externals are more lateral and their fascicles are going down and in. Our internals are more medial and their fascicles are going down and out. So even though for this one, we obviously can't see which one's superficial and which one's deep, we can clearly see other characteristics that let us tell which one is internal and which one is external. Now, not surprisingly, being between the ribs, their job is going to help us to move the ribs, help us to play an important role in breathing. And what are the two breathing processes we just identified?
Inhalation.
Inhaling and
And
exhaling.
exhaling.
One begins with an E, one begins with an I. So wouldn't it be super convenient if our internal intercostal inhaled and our external intercostal exhaled?
Yeah, the
Wouldn't that be
hiatus.
super, super convenient?
Yeah. It would.
Yeah.
Yeah, but does it?
No.
No, it's actually the opposite, which also makes it easy to remember. The external inhales, the internal exhales. But basically what happens is the externals bring the ribs up for our inhalation and our internals bring the ribs down for an exhalation. So these two are additional accessory muscles that help in the breathing process. All right. Questions on those? All right. Good news is there's only one thing left we need to cover to make it through our trunk, right? Just 4 more muscles.
Yeah.
Yeah. These 4 muscles form a muscle group. And this muscle group happens to be called the abdominal girdle.
Do we need to know that for all of them?
Yep.
Okay.
And how many muscles make up the abdominal girdle muscle group?
4.
4. Again, remember, as we learned in the bones, a girdle is a support structure, something that holds things together. There are no bones protecting most of the muscles of our abdominal pelvic cavity. So instead, we have to have multiple layers of muscles. And those multiple layers of muscles are all at different orientations to provide basically this mesh-like support structure that provides that protection. And I think we talked in this class about the example of Houdini, right? Getting punched in the stomach, not being able to prepare versus being able to engage those core muscles. When we talk about engaging those core muscles, that's what we're talking about, the abdominal girdle muscles, and there are 4 of them. So let's identify them by their orientation. The first of these are ones that happen to run vertically. Long, straight muscles, and they're paired, so they're on both sides. And what is this long, straight muscle?
The rectus abdominis.
Yeah, remember rectus means straight. It is in the abdominal region. So we have this rectus abdominis. Now, remember, we need these muscles to protect because we don't have any outside bones. But remember, for bones to move our body through space, they do have to relate to bones. What bones do our rectus abdominis muscles relate to? What bones are they affecting? What bones are they moving?
The sternum?
Not a bad guess, but again, they're below the sternum, so it wouldn't necessarily be the
Or
sternum.
vertebrae?
Vertebral column?
Vertebrae, exactly, absolutely. Basically, what it does is it affects the vertebrae. Notice our rectus abdominis are parallel to the vertebrae. So are they going to pull our body to the side or twist it or turn it or anything?
No.
No. So how many actions does our rectus abdominis have?
One,
One,
two.
two, right? What's the one?
Lateral flexion?
Well, remember, it's not lateral flexion, but just flexion
Flexing
coming
the
forward,
vertebral
right? When they
column.
contract, they bring the vertebral column forward. So they do a flex, right? However, remember, as we mentioned, we need these muscles for protection. So when we contract these muscles, all of this abdominal girdle basically forms a circle around our abdomen. And when we contract a circle, what happens to the lumen of that circle?
It gets smaller.
It gets smaller, right? Again, we all know this because I'm sure, like me, when you're standing on the beach, hanging out down on the beach, and you happen to see the Jonas Brothers walking by, what's the first thing you do to impress them? Suck in the gut, right? Compress that abdomen. You compress that abdomen to tighten the muscles, to decrease the size, and so you can look cuter for a Jonas Brother, right? So there you go, compressing the abdomen and flexing the vertebral column. That is our rectus abdominis. Now, one of the interesting things about our rectus abdominis, as I'm sure you know, is that it is divided by tendons, tendinous insertions, into not one, not two, not four, not six, but 8 individual bellies. Yes, Amanda.
The abdominal girdle muscle group isn't on our list. And I see rectus abdominis, and then you said minus. So I'm just trying to see if they're the same or.
Yes, singular versus plural.
Okay. And then.
But yes, again, remember, you are this is a muscle group where you're responsible for all 4 individual muscles. So it's important to know that they are a functional group, the abdominal girdle, but we definitely are responsible for each of these individual muscles. So that's the emphasis on this one. Okay.
I think she's like asking if we're responsible for knowing the name of it, of all 4, like the
Yes,
group name.
you are responsible for the name of the abdominal girdle. If I asked you to identify the 4 muscles that made up the abdominal girdle, you would need to know what that meant. Okay. All right. 6 tendinous insertions that divide the rectus abdominis into 8 individual bellies, which we commonly refer to as what?
Abs.
Yeah, your 6-pack, or what is really an 8-pack. Absolutely. What we commonly think of as a 6-pack is really an 8-pack. I think, actually, I don't know if I have it going forward, so let's just cheat and go back. There we go. Right. Notice here in our bodybuilder, we see the 1, 2, 3, 4, 5, 6, 7, 8 bellies of the rectus abdominis muscles, that 8-pack. One of the interesting things is that each of these bellies is individually innervated, which means it can be individually controlled. Now, just because it can be individually controlled, does that mean that everybody has that control?
No.
No. The same way that all of us have elevators to the ears. Every single one of us has elevators to the ears and nerves that control it. But can we all consciously wiggle our ears by being able to contract that muscle independently?
No.
No, and it's the same thing here as well. If you've ever seen his YouTube videos, Terry Crews is a great example of an individual who actually has that control. He actually has electronic tensors set up to different parts of his body so that as he contracts those parts of his body, they produce an electrical signal and it plays a musical instrument. And so if you've ever seen his YouTube videos, he actually plays music by just contracting the different muscles of his body.
So why do some people have that control? Like I know people who wiggle their ears.
Yeah,
I don't know.
who knows?
Who knows?
Is it,
Okay.
you know, if we maybe it'd be something that if we all sat down, we'd be able to learn. You know, why?
Yeah.
Why I don't have a good answer as to why some people have that conscious control and other people don't. Maybe it's something that can be learned. Maybe it's genetic. I just know that there is that variation. All right. Excellent. So again, back to here. Perfect. It is a superficial and it is the most medial muscle. So we should probably emphasize that on our picture as well. These are the most medial of all of our muscles. Notice laterally and deep our horizontal muscles. And what do we call the horizontal muscles?
Transversus abdominis.
There you go. This is our transversus abdominis. Excellent. And it is, again, a horizontal muscle. This is our deepest of our abdominal girdle muscles. And notice also its orientation. With all of these fascicles basically going horizontal, do they really cross from one vertebrae to the next?
Oh.
No. Essentially, basically the transversus abdominis muscles from the two sides basically just make a circular muscle around the abdomen. And since it doesn't cross different levels of the vertebrae, how many actions does our transversus abdominis have?
One.
Yeah, just that squeezing, that compressing of the abdomen. All right. Questions on that? Now, remember up here in the ribs, we had an external intercostal and an internal intercostal. What are the two remaining muscles that we have?
Internal and external obliques.
Excellent. We have an internal oblique, and we have an external oblique. Let's start easy. What do you think the biggest difference between the internal and the external oblique, or one of the biggest differences between the internal and external oblique is?
Their location.
Right. So external is what?
Posterior.
True. In some places it could be posterior, but in other places it could be
Deep.
anterior or lateral. So there you
Yeah.
go. It is superficial. Oh, helps if I spell superficial, right? It is superficial, just like the external intercostal. Also, like the external intercostal, its fascicles project down and in. Remember, in our intercostals, the externals are more lateral. Well, here in the oblique, our externals are more superficial, are superior. They actually cover the ribs and actually go all the way to the serratus anterior. So let's cheat and put this down below so that we can then draw our external obliques. And of course, remember on this side, they're going to go down and in as well. And that brings us to our internal oblique. As the name indicates, these are going to be deep. Guess which way their fascicles project? Well, which way did our internal intercostals project? I am speaking out loud, right? There we go.
Down
Well,
and in.
remember, our externals are down and in. That means our internals are going to be down and out. And this is going to be more inferior. Does not go over the ribs. So, oops. Notice in this fashion, we have all these muscle fibers going at all these different orientations. Let's, now that we've drawn this, look at the pretty pictures from your textbook. Notice here, we can see, here is your external oblique. Notice its fascicles are projecting down and in. Notice it's coming up over the ribs and it interdigitates with the serratus anterior. Notice on this side, they've peeled it away. So here we have the internal oblique that only goes up to the ribs and its fascicles are going down and out. And then deep, we have those transversus abdominis. It's nice how they've shown us here, but remember how with our intercostals, they were able to pull all of them apart and show it to us separately? And it's a great way to see it. They can do the exact same thing in your textbook for, oops, where is it? There we go. For them individually. Notice now we can clearly see each one of these independently. Our vertical rectus abdominis with its tendinous insertions forming the four bellies on this side. Notice our external oblique comes up over the ribs and its fascicles go down and in. Our internal oblique does not go over the ribs and its fascicles go down and out. Our transversus abdominis is the deepest and its fascicles are horizontal. Questions on the identification of these muscles? Their location, their orientation, anything on the anatomy of them? All right, excellent. Then let's go back to the picture because we have one minor issue we have to deal with. Notice something. The internal oblique on the left side of the body projects the exact same way as the external oblique on the other side of the body. The external oblique on this side of the body projects the same way as the internal oblique on the other side of the body. So when it comes to their actions, what do you notice about the internal and the external obliques?
They'll have the same actions probably.
They have the exact same action because when you're actually doing these actions of twisting or turning or anything along those lines, when you're doing that, you're actually going to use the internal oblique on one side and the external oblique on the other. So their actions are essentially identical and give them to me. What are the 4 actions of these muscles?
Compressing your abdomen,
All right, sucking the gut in, compressing the abdomen. What else?
lateral flex,
Lateral flex. What else?
flexing,
Flexing and
rotating your vertebrae.
yeah, rotation. So rotation, lateral flex, flex, and compressing the abdomen. All right, and those are the 4 actions of both the external oblique and the internal oblique. Now, as I mentioned, I really like this picture from your textbook because it shows the muscles individually, but notice it shows something else. You are not responsible for the origins and the insertions of the abdominal muscles, but notice there is a structure here you are responsible for. There is a ligament that goes from the superior anterior iliac spine to the pubic tubercle. And what is this structure you are responsible for?
Inguinal ligament.
It is indeed the inguinal ligament. We see it really nicely on this illustration, but even if we go back here, we see it really, really nicely. Again, from the superior anterior iliac spine to the pubic tubercle. I know that pubic tubercle seemed like a silly little bump on the head of the pubis you needed to know, but now we know why. It's because where the inguinal ligament connects. And the picture I really want to show you is this one. Again, here we see that inguinal ligament, but in boy type people, there is a structure that actually passes through this inguinal ligament and comes out. Here we see it. And it forms an opening called the inguinal canal. This structure that comes out is the spermatocord. What does the spermatocord do?
Can I guess?
Yes,
Is it where sperm goes through?
absolutely. Where is sperm produced?
Balls. I don't know.
Okay. Testicles would have been
Testicles.
probably a more appropriate answer, but balls work, I guess. Absolutely. And where are those testicles located? Come on, Ava, can't be the only person who's ever seen these before, right? They are located outside the abdominal pelvic cavity, right? To regulate temperature. And then the sperm have to come back into the body. Of course, making sperm is an elaborate process. So there are blood vessels going to and from. And as anybody who's ever won $10,000 on America's Funniest Home Videos knows, right? There are lots of nerves that go to the testicles as well, right? To those testes as well. So all of those structures need to pass back and forth from the inside of the body to the outside of the body where the testes are located. And that's the function of the spermatocord. Are the female ovaries located outside of the human body?
No.
No. Females also have this inguinal ligament, but their canal is a potential space, right? Potential space. When I put my 2 hands together like this, do they fuse together to become one thing?
No.
No. Is there anything between my hands?
No.
No, there's nothing between them, but they're not fused together. So there is a potential of a space here between my hands, but it's not there right now. And that's what I mean when I say there's a potential space. In a female, this inguinal ligament is basically closed. There's nothing that passes through it, so it's just a potential space. Where in males, it's an opening that something passes through. Well, why is it significant? Why am I making a point of emphasizing this? Because here in the abdominal pelvic cavity, if the pressure gets too big, you're trying to lift something too heavy, right? Or some other type of very vigorous activity is taking place where you have a very high increase in pressure in your abdominal pelvic cavity, that pressure will try to find a place to escape. And when it finds a place to escape, it can actually lead to a bulging as fluid or even some of the organs of the abdominal pelvic cavity are pushed out of that opening. What do we call that condition?
Is that a hernia?
That is exactly what a hernia is. Now notice, males, an inguinal hernia is quite common. Do females get inguinal hernias?
I don't think so.
No, it would be very, very rare. I can't say that it's never happened because it is a potential space, but it is very, very rare. However, is there a location where both males and females are equally likely to get a hernia?
Yes.
Yes.
Where would that be?
In the abdominal wall.
Yeah, and specifically, typically at the belly button. Right, the belly button is another weak part in there, and since everybody has belly buttons, everybody is equally likely to get, both males and females are equally likely to get an umbilical hernia. So that excess pressure forcing that fluid or forcing those organs out of those weak spots, we call a hernia. And that inguinal canal makes males much more susceptible to a hernia in that inguinal region than females are. All right. Notice one last thing while we're looking at, well, actually not last thing. So the inguinal ligament is one of the structures we're responsible for. Notice there are a couple other structures we're responsible for as well. One of those, as we see on the illustration here, is the big, broad, flat tendon of the external oblique. And what's the name we have for a big, broad, flat tendon?
Aponeurosis.
Yeah, it's the aponeurosis. This is the aponeurosis of the external oblique. What's interesting is all three of our lateral abdominal muscles actually have aponeuroses. Your book does a really great job of showing this in this view, a cross-section view. Notice in this cross-section view, we see again that aponeurosis of the external oblique that is on top and goes over the top of the rectus abdominis muscles. But notice deep to that, both the internal and the transverse have aponeuroses. But do we see that from the front view?
No.
No. What we do see is where all these collagen fibers come together. Notice we have the collagen fibers of all three aponeuroses from one side and all three aponeuroses on the other side that actually all fuse together to form a white line right down the center of the body. Right? Notice we see that here, and we can also see it when we go back to this picture. This white line is where all of those aponeuroses, collagen fibers fuse together. And what do we call that white line?
Linea alba.
There you go. It's Jessica's younger sister. Right? Linea alba. Linea, of course, means line. Alba, white. It's the white line. We talked about some of the fun changes that females' bodies will undergo during pregnancy. As that belly is expanding, one thing that will sometimes happen is you will get a thinning of the collagen fibers of the linea alba, especially inferior to the belly button. And as they thin, you can kind of see, I mean, not that you're going to see the baby moving around or anything, but because the wall thins, it becomes darker in color and is known as the linea nigra. Right, because that's that dark line that forms typically after birth, and everything starts to recoil back. That typically goes away, but it's not an uncommon feature to see in pregnant females. All right, so those are structures you need to know. There's one last structure you are responsible for on the torso, and that is this structure, this posterior structure. And a lot is in a name. This is the thoracolumbar. Thoraco, of course, refers to the thoracic region. Lumbar means the lumbar region. But notice this is not an aponeurosis. This is a fascia. Remember, an aponeurosis is a tendon from a muscle attaching to the bone, anchoring it in place. Remember, as we talked about, fascias are connective tissues that stabilize multiple muscles. Here we see, for instance, the latissimus dorsi, but there's also a bunch of muscles that move the spinal column that are all stabilized and held in place. So this is a sheet of connective tissue that wrap around all of these muscles, which is why it is called a fascia instead of an aponeurosis. So on the posterior side, we have the thoracolumbar fascia. On the anterior side, here we see it, but also here, what if we go all the way back to, where was that picture? Here, this is the one I want. Here's that chart. And again, notice on our chart here, we see both aponeuroses of the external oblique. Notice we see the inguinal ligament, and this one actually has inguinal canals drawn in them. And we can see the linea alba that goes right through the belly button. So notice, because our chart just shows us the superficial things, we can very nicely see all of those features really nicely here on the chart. All right. I think that's all the muscles and all the structures for the torso. Did we miss anything?
No.
All right, excellent. Then even though it's 15 minutes early, I don't see a point in starting on the arm. I think we can save that for the next class. So how are we doing on time? We have 3 more classes. Perfect. We're doing perfect. We're doing awesome. So excellent. That's right where we want to be. And so I think that is all we need to cover for today. All right. Questions on any of that?
When we're studying for this material, I was thinking about ways to approach it. So I was going to make flashcards. I don't know about anyone else, but would you suggest us learning where the muscles are first, like looking at the charts or looking at our atlas and learning where the muscles are before making flashcards? Or do you do it, or should we do it the other way around, where like we learn the origin, the insertion, the action, and the muscle first?
I think because of the way you're going to be tested on this material, I think it is worthwhile to look at all of them together. And remember, like we talked about last time, make a 4-sided flashcard. But here's the key to your 4-sided flashcard, right? Again, you take your normal index card and you divide it in half. And so, like I said, on one side, you're going to write origin, and you're going to write whatever the origin is. On another side, you're going to put insertion and whatever the insertion is. On the other one, you're going to put action. And the last muscle we just finished talking about was the external obliques and the internal obliques. So let's say 4 actions to be specific. And you're going to say what those are. So you're going to do all of that. You could, on this 4th one, you could put just the name of the muscle and have that there. Or a good way to test yourself is actually to take, I've provided you with unlabeled pictures. Take a picture and take a picture of the body, right? And on the body, have that body. There's the body, of course. Here's the head, of course. And then on it, you can have an arrow pointing to that muscle that is coming down and in on the left-hand side. So now as you look at this picture, you have to think, all right, that is the external oblique. I'm recognizing it on the picture. And then I can think to myself, all right, what is the origin, or what is the insertion, or what is the action? And then when I'm looking at it the next time, I can think, all right, what muscle originates? Now, obviously, this one isn't one that has an origin, so it doesn't work for that. But you get the idea of what I'm saying. Show a muscle and then try to think of what it is, and then try to visualize where it is on the body. So being able to put these 4 pieces of information together, where a muscle is located, what its origin is, what its insertion is, what its actions are something that can be really helpful for putting it all together. Now, notice if it's like this one, where you don't need the origin insertion, then you just need a normal 2-sided flashcard. Picture on one side, actions on the other. Okay? So yeah, but for the ones with the origin insertions, 4-sided flashcards work well. For other ones, 2-sided. And again, it's your learning style. Flashcards help me because I I'm a tactile learner, so actually taking the time to write them out is something that really helps me. And I'm a visual learner, so taking the time to look at them helps me to recall that. Not everybody learns that way. So if you are more of an auditory learner, then that's why we're recording the lectures. Keep listening to them again and again and again and again, right? Or if you learn by writing things out, then rewrite the lecture slides. Whatever helps you to be successful. There is no one way to correctly study for this. There's just the ways that help you to be successful. And everybody's going to have a different style that's going to help them to learn this material. Okay. All right. Awesome question. Any others?
I had one more. If anyone wants to go first, you
Go
can.
ahead.
I'm sorry.
Angie has one. She can go first.
Yeah.
Oh, okay. Okay, so to build on that, so I know on the lab exam, the bolded muscles would be, you would ask what muscle is this origin or this insertion. For the ones that aren't bolded, then you would just ask, identify this muscle. Is that how you would word ones that don't have insertion and origin?
Okay, so here is a picture that I guarantee is going to be on the exam. Okay. I guarantee this is going to be on the exam. This is something in the classroom. This is absolutely positively going to be here. So let's do something that we've already done. On the exam, I could have an arrow pointing to this muscle right here. Maybe I'll change the color of it so it stands up a little bit better. All right. With this picture and that arrow, how many questions can I ask you?
4.
Yeah, what are they?
What's the muscle, or what's the name of the muscle?
Exactly. So hold on. Identify the muscle. What else?
Identify the origin.
What else?
Identify the insertion of the muscle.
And
Identify the action.
2 actions, because this one has 2 actions. 2 actions of this muscle.
Mhm.
All right. 4 questions that I could ask with this. However, on the other hand, I could also use this picture. I could also have an arrow, and I could have that arrow pointing like this. And how many questions could I ask there?
2.
2. What are they?
Identify the muscle.
And
Action.
4 actions.
Or action.
Muscle. Remember, I will always give you, for the actions, I'll always give you the hint of asking for the precise number of actions for a muscle. All right. So here's a great example. If this green arrow, you can't tell for 100% certain whether I'm pointing at the serratus anterior or the external oblique. If I'm asking for 4 actions, which one does it have to be?
The oblique.
It has to be the oblique because the serratus anterior doesn't have 4 actions.
Okay.
All right.
Okay. Thank you.
One more. Since we're here, this is not the best view, and I guarantee you I will not do it this way. This is merely for educational purposes. How many questions can I ask here?
5.
No, 2.
Oh.
What are the 2 questions?
Identify the structure.
Okay, see, that's why I told you it wasn't. Let's pretend this is pointing to the muscle.
That's why I was confused.
Obviously, I would ask you to identify the action, but the second question I would ask, identify the muscle, be specific. What would your answer to that be?
Occipital frontalis, frontal and occipital bellies.
Am I pointing to both of those?
No.
Frontal, sorry.
There
Frontal
you go,
belly.
exactly. And that's what, and again, notice you showed tremendous knowledge with that answer. You pointed out the muscle, the occipital frontalis, and you pointed out that it had two bellies, and you correctly named those two bellies. Everything you said in that statement was true. But if you did that on the exam, I could not give you full credit for it. Because here I'm asking you to be specific. Which specific belly am I pointing at here? So for this, you would say the frontal belly of the occipital frontalis. And like I said, this is not the way I'm going to show the frontal belly of the occipital frontalis. This is not a good view for this. I just, while we were on this page talking about the questions of the exam, I wanted to remind you about that be specific, right? We'll do the exact same thing when we're talking about the arm. Notice as we look at the bicep brachia here, there are two bellies to the bicep brachia. These two bellies have two different names and have two different origins. And so you need to identify them uniquely. And again, I will use that magical phrase, be specific. When you see be specific on this exam, warning bells should go out that I am looking for a specific belly or a specific origin. That's what be specific means. That means give me one precise origin, or it's going to be one precise name. So when you see that term be specific, that is what be specific means. Give me this precise name of this one belly, or give me the precise origin of this one belly. Not all of them, just the one I'm pointing at. That's what that be specific is going to mean. And so these questions that you see here on the screen, gosh, that's probably 75, 80% of the lab exam. Basically, these 5 questions: identify the muscle, be specific, identify the origin, identify the origin, be specific, identify the insertion, and identify X actions. And that's the vast, vast majority of the exam.
So I had a question similar to Angie's, because when I was looking, for example, if we're looking at the trapezius and the rhomboids, if you asked us, you pointed to the spine and said, well, what's the origin? They both, depending on where the arrow points, could be two different things. So would you like show, like if we're doing the bones, for example, would you show like a bracket of what bones? That was my...
No, and that is an awesome question. So it gets trickier when we get to the, well, it gets, trickier is not the right word, more obvious when we get to the, here, let's do it this way. We haven't done any of them yet.
I can wait too. I was
No, it's okay. No, I want to, since we have a few minutes, I like bringing this up now. So from what we've covered so far right now in this class, and today actually for that matter, on the exam, I could have this bone with an arrow pointing to that bone feature and ask you to identify the muscle that inserts into that bone feature. And what would the answer to that be? Anyone remember what inserts into the coracoid process of your scapula?
Is it the trapezius?
Not the trapezius.
Pectoralis minor.
Pectoralis minor, absolutely. However, this is also going to be an origin point for another muscle. So I could have this exact same picture with this exact same arrow, and I could ask you to identify the muscle that originates from this bone feature, and I would actually say be specific. And does anybody know what the answer to that is? It's okay if you don't, we haven't gotten there yet, but.
Sternocleidomastoid?
Not one we've covered yet.
Oh.
It is and partial credit for the bicep brachia, but again, I said be specific, so you would actually say short head of the bicep brachia. The short head of the bicep brachia actually originates there. I could do the same thing by taking this arrow and pointing it towards the spine. Today, we identified a muscle that inserts into this bone feature. What is the bone feature that inserts into this? What is the muscle that inserts into this bone feature?
Trapezius.
Trapezius. And as we'll learn, the deltoid actually originates from it. So I could ask that question that way. The other thing that I can do, and again, it's not fair because we haven't done any of them yet, but what bone is this?
It's the humerus.
It is indeed the humerus, absolutely. And on the humerus, there is an indentation right here. Anybody remember what that indentation is?
Enter.
Yeah, the intercholeric line.
Condylar.
Close.
Anterior.
Intertubercular groove or sulcus, right? It's between the tubercles. As it turns out, there are actually two muscles that insert into this. So on the exam, I may have an arrow pointing right here, and I would say, identify the two muscles that insert into this region. When I get to the tibia, which we won't do till next week. There you go. What muscle, what bone is this?
Tibia.
Tibia.
Tibia, I just said it. There you go. That's excellent. It is indeed the tibia. It wasn't a trick question. It is the tibia. Excellent. This underneath the medial condyle of the tibia, right here in the medial condyle of the tibia, there are four muscles that, oh, I should have used blue. There are four muscles that insert into this bone feature. So on the exam, can I point an arrow at this underside of the medial condyle and ask you to identify all the muscles that insert into it, or all four muscles that insert into it?
Yeah.
Yeah. There's also four muscles that insert into the tibial tuberosity. So we could do that as well. Right. And
Basically, all the origins and insertions are pretty much going to be on the bones.
right. So yes, so any of the origins and insertions I could hold you for on this. And if you're ever in the classroom, you'll actually see the skeletons in the classroom actually have blue and red on them. There's some real skeletons, and all the plastic skeletons have the origins and insertions on them. So you can see where these things are. So over here is an origin, four muscles insert here, four muscles insert here. There's a lot going on in the tibia. So, so yes, so if an attachment point is for more than one bone, then I will ask for more than one muscle, then I'll ask for that. Identify the four muscles that insert into this bone feature. Or, like I said, if it is an attachment point that can be both an origin and an insertion, then I could ask that both ways. I could, on the exam, have an arrow point here, and question one could be identify the muscle that originates from this bone feature, be specific, or identify the muscle that inserts into the bone feature, right? Just like I did on the spine, just like I can do on the chromium. So yes, some of these bone features are shared by multiple muscles. Some it'll be a shared attachment point, like two origins or two insertions. And sometimes what we say is the muscles embrace. If one muscle inserts where the next muscle begins, originates, we say that those two muscles embrace. And so as we start moving into the arms and legs, where we have a lot of muscles and very few bones, there's going to be a lot of sharing of attachment points. And so we're going to see that as we move on to the appendages.
So then
All right.
really studying the bones and applying where the muscles would be more beneficial, because that's mainly what some of what the exam is going to be. Because if we study just the muscles, let's just say on like because most of the muscles, like the diagrams and stuff, they don't include the bones usually.
Correct.
They just.
But so
That's true.
the two things that I will remind you is, remember, this is a cumulative class.
Right.
So the things that we learned in the bones, we did for a reason. I didn't just randomly put all the bone features in a hat and pulled them out. I picked bone features that were going to be important, things that we were going to use again. So this is a cumulative class with a cumulative final exam. So you can't just purge all that information after you've taken the class and forget about it. And I wouldn't say that a majority, obviously you need to know the bones, you need to know the origins and insertions. But remember, I said probably somewhere, 15 to 20% of this lab exam is going to be bone pictures. So again, it's not a huge part of it, but is there going to be a picture of the scapula on the exam? Yes, I absolutely guarantee you, because there are so many attachment points, right? As we'll see, there are muscles that sit in these fossas that we've learned. There's a massive amount of stuff that can, muscles that are related. And in fact, being the smart, clever, sophisticated students that you are, if you were to look at the handout of the muscle models, you will actually see that one of the models that you're responsible for is indeed the scapula and the muscles that are associated with the scapula. So you've got 5 days till we meet again. There's no reason you can't start thinking and looking ahead at this material, because this is stuff that you are going to be responsible for. All right. Excellent. Questions on that? All right. Study hard this weekend. Again, you only have 2 weekends left to study for this exam. Is that right?
Yeah.
Yeah, 2 weekends. There you go.
Yeah.
Only 2 weekends left to study for this exam. So busy time. All right. Good luck. Have fun.
Thank you.
Bye.